Hello to all my wonderful children. A very good afternoon since it is sharp 12. I can clearly say a very good afternoon. Let me see if at all I'm audible and visible. Yes, I am. Hello Ahad, hello Shreya, and hello, it's Janu. Very nice name. Okay. Hi guys. How's the Josh? I hope every one of you is doing good. Yeah, we're all busy studying a lot of things, right, for boards. Hello Nandita. Good to see lots of new children out there. Hi ma'am, I'm Swaroop. Hello Swaroop. Paru is also here. Hello Paru. Okay, so we'll just wait for a minute so that everyone else can also join. And please send this video link to all of your friends who are there, who really want to go through model question papers and needs a company also. So I'm going to give them a company. Yeah. Just a second. Prithvi, oh my god, Sudarshan, so many people are there. Vishnu and Klasi, Gokuleshwar, hello, hello, hello. Shrivar, Shazam, yes, I am doing well, thank you. So I was little down with some fever and little cold. I think that's why my voice is also bit changed, but now I'm fine. Thanks for asking. Okay. So let's go ahead. Shall we go ahead and solve all the model question papers? And make sure you send this video link to everyone who is struggling with model question papers. And Vishnu ma'am, how much could I score if I follow section wise analysis? Section wise analysis is what do you mean by that? What was I saying? What do you mean by that? No ma'am, voice is clear. Okay. My voice is clear, right? Yeah. So, do you mean like all 1 marker, all 2, 3, 5 marker, 2, 3 marker and all 5 marker questions? Are you trying to ask me about that? Ma'am, model question papers are so important. Yes, it is. It is definitely important. I'm telling you for everyone before I start ahead with this particular video, one very important thing. Questions might not be repeated from the model question paper, but the concepts will. Okay, so make sure you know the concepts really well. That is our ultimate aim. Okay, and yes, if at all you go ahead with all the 1 marker, 2, 3 marker and 5 marker videos that I have done in this particular channel, it will be more than enough, because in that also I have covered all the model question paper questions. Yeah, perfect. Let's go ahead with the first question that we have, and there it goes. Try Madi, everyone. Okay, statement 1: pollination does not guarantee the transfer of right type of pollen. Statement number 2: compatible pollen pistil interaction guarantee double fertilization. What do you think is the answer for this question? You have the options right over here. Ma'am, how much time will this particular lecture continue? Until the model question papers are over. So if at all you give me answers quickly and you understand concepts quickly, then what is going to happen? It will end up slow. It will end up fast. Otherwise, it might take around like 5 hours, I think. Yes. Sudarshan says the answer for this question is A. Okay. Prithvi says the answer is C, B. Okay. A, B, C, D for me. But maximum answers, Neha, Patil, Sudarshan, everyone says it is A. It is A. So which is why I'll lock A and I'll see if at all it is correct or not. Okay. I have to check it. So pollination does not guarantee the transfer of right type of pollen. Yeah, Baba. Why it is not guaranteed? I have told you in the previous videos of 1 marker. Suppose a pollen grain came from some other plant. This can be any plant. Pollen grain can land on it. It is not necessary that it is of the same species and things like that. So this particular thing is correct. Pollination does not guarantee the transfer of right type of pollen. Now statement 2 says compatible pollen pistil interaction guarantee double fertilization. Do you think that is true? Do you think that is true? For those of you who said A, you have to think twice. Compatible pollen pistil interaction. Suppose over here everything is going on really well, and we say pollen tube is also formed. Do you think that double fertilization will definitely happen after this particular course? No, Baba, you cannot say that. Always understand this very important thing. That is, multiple pollen grains can land on the stigma. Multiple of them can form what the pollen tube. Only one might be able to go ahead and do the fertilization. We do not know. We do not have any guarantee on that. That fertilization will happen, will not happen. Pollination does not guarantee fertilization. And also, compatible pollen pistil interaction also to some extent it does not guarantee. Why? Because even if it's compatible, even if it's making pollen tube, not all of them will be reaching things out there, right? Which is why I'll say I cannot totally go ahead with the statement number 2, which is why I'll say statement 1 is correct, but statement 2 is what? Incorrect. In that particular condition, your answer is going to be what? C. Your answer is C. Let me see who all gave me the answer C. Prithvi gave me the answer as C. Okay, Prithvi had a different answer before. Paru gave me the answer as C. Yeah. Perfect. Okay, perfect. Ma'am, please do one short video on the MCQs. Dear, I have done it. All 1 marker videos there. Okay, ma'am, we will get break, right? Yes, you will get break. I'll also get break. Perfect. So all of you, everyone who understood this particular question, can you just give me a thumbs up? Ma'am, Google is saying answer is A. Don't trust Google. Trust your NCERT. Go read your NCERT. Okay, guys, understood the question? Can I go ahead? If yes, just give me a thumbs up. We'll quickly go ahead. Hello, Gia. Hello. Okay, perfect. So, guys, I will go ahead. The answer for this question will be C. Next question that we have right over here is, the secondary oocyte formation from the primary oocyte occurs in which of the follicle? Thank you. Okay. The secondary oocyte formation from the primary oocyte occurs in which of the follicle? Primary follicle, secondary follicle, tertiary follicle, or Graafian follicle? So you are being asked. Answer is C. Sudarshan says C. Dhanush says C. Prithvi also says C. Okay. Paru also say, oh my God, lot of children are saying, I have read out all your names, okay? All of you are saying C. Is C the answer? Let me see, okay? So first of all, you have the primary follicle, Baba. First thing that you have is the primary follicle. What happens inside the primary follicle? You have the primary oocyte, and just surrounding it, you have some granulosa cells. Okay, the primary oocyte is stuck in a stage. What stage? I'll say it is stuck in meiosis stage. Okay, in meiosis first. In meiosis 1, everyone knows that in which stage of meiosis is it stuck? It is actually, anyone who knows the answer, you can tell me right now. In which stage it is stuck as of now? It was undergoing meiosis 1, but it is stuck. Now the second thing that happens is you have a secondary follicle being made. What happens in the secondary follicle? The primary oocyte remains the same itself. Okay, you had granulosa cells, but now you will be having some extra granulosa cells. Apart from that, a new theca is also being formed. A new theca layer is being formed. But what remains inside? It's the same primary oocyte only that stays inside. And yes, someone has given me the answer. Vishnu gave me the answer. It is stuck in what stage, Baba? Prophase 1. Prophase 1. So in meiosis 1, you have prophase 1, metaphase 1, anaphase 1, telophase 1. It is stuck in prophase 1. Now what happens? Now you have tertiary follicle being made. What happens in tertiary follicle, Baba? You see, this particular primary oocyte which is there, it is finally going to undergo meiosis. It is going to finally undergo meiosis, and you are going to get two things. One is your secondary oocyte. Apart from that, you have your first polar body being made. And also you would be having your granulosa cells. The cells which are there, they will be still over here. You will be having a cavity in between. This is known as antral cavity, or you also call this antrum. Okay, and what about the theca layer? The theca gets differentiated into inner layer and outer layer. Inner layer and outer layer. But finally, you get secondary oocyte over here. So until now, you just had primary oocyte, which was 2N. It underwent division, that is meiosis 1. It is unequal division. One cell will be big, one cell will be small. This cell which is small is the first polar body. This cell which is big is known as the secondary oocyte. Okay, and this happens in the tertiary follicle. So your answer for this question is going to be C. So everyone who said C, perfect. Okay. Oh my God, so many questions are there. Guys, I'll take your questions one by one. Okay, just give me some time to read your questions as well. Now I'll be showing you the next question. You tell me the answer. By that time, I'll read your questions. Progenies with AB and O blood groups are not possible from which of the following mating parents? That means AB and O blood group progeny should not be made. Then what should be your parents' genotype? That is the question over here. So you have to give me the answer for this. And by the way, let me read out everyone who gave me the correct answer before. So Sudarshan, ancient India history. You didn't miss anything there. Dhanush and Prithvi and Paru, Rakshit and Shreya. Awesome. Deep Nevi and Dinesh, Jia. Oh my God. Kids Zone. Perfect. Every one of you are doing absolutely great. I have Lakshmi over here. By the way, I want to tell one very important thing. If at all I miss reading out your name, please forgive me. So many names are popping up, right? So I have all of you in front of me, and I'll really cater that. Love you lots. Okay. So your answer for this question is A. Most of you guys are saying A. Okay, let's see what happens in A. A is basically O blood group and AB blood group. For a person to have O blood group, Baba, your blood group should be what? IO. Because O is only seen in recessive condition. That means both the alleles should be O only. Fine. What about AB? A and B are co-dominant, which is why it should be like this. Now, whenever you do the crossing, what do you get? You get IAIO over here. You get IBIO over here. Okay. you get again IA, IO over here, and finally again you get IB, IO over here. So whatever you do, A is dominant over O, which is why A blood group, B is dominant over O, B blood group, A is again dominant over O, A blood group, B is dominant over O, B blood group. Did we get AB or O blood group? No, which is why your answer would be this. Now how will you get to know that others are not the answer? Listen to me very carefully. For a person to have AB blood group, they should receive one allele from one of the parent, that is at least A allele, and from the other parent they should be getting at least what? B allele. So if at all, this is the condition. So it should be made sure that in opposite side there is no A and B together. For example, here I have A, but here I also have B. That means this A and B can come together and give me AB. Similarly, here I have A, here I have B. Again, this A and B can come together. Here I have A, here I have B also, and here I have A. This A and B can come together. You get it? Which is why your answer cannot be all this thing. Your own answer can be only A. Fine, perfect. So all my dear champions who gave me the answer, you are doing absolutely great. Next question of model question paper. Another chemical name for nitrogenous base thymine is. What is the next other name for thymine? Now, ma'am, okay, Lakshmi, ma'am, if I read model and preparatory paper, can I get how much mark? Lakshmi, you have to listen. You have to know one thing. Model question paper solving would only be enough if at all you know the concept behind each and every question out there. And also, you have to go ahead and solve the question bank questions, which is why I've already done videos on 1 marker, 2 marker, 3 marker, 5 markers separately. Watch those 3 videos, that would be more than enough. And yesterday's was recorded session, but you didn't read our comments. So yesterday I had a parallel live somewhere else also, and as I said, my health is not that great. Today I'm a bit okay, right? So yesterday I was not that great, so I went home and I slept. Okay, I'm so sorry, but yeah. Yesterday's recorded session was really helpful. Thank you, Jia. Perfect. So you guys are giving me the answer D. Ma'am, where can I get the 5 marker? Okay, I think it should be out. Actually, I recorded it yesterday only, but for some reason it was not. Okay, it is still not published. I'll just check with the team, okay? You will get it. Don't worry. Today only I'll make sure that you get it. Okay. Perfect. So all my dear children are giving me the answer for this question as D. What is D? 5-methyl uracil. Is thymine known as 5-methyl uracil? Yes, Baba. It is known as 5-methyl uracil. So who came first, uracil or thymine? Listen, in your RNA, okay, in your RNA, you have A, G, and instead of T, you have uracil and you have C. So this is something that is different in the case of DNA. In the DNA, you have T. And this T, when you have it, it is more stable. This makes your DNA very much stable. Why? What is the problem with this? Uracil is not something that is more stable. So what do you do to make it more stable? You add a methyl group on the 5th position. So how does it look? It looks like this. At the 5th position, Baba, you are adding a methyl group, and that's how you get a stable thing that is known as, what do you call, thymine. So your answer for this question would be what? 5-methyl uracil. Do not, people remember methyl, they forget 2 or 3 or 5. So please remember it's 5. 5 star. Okay? Yes, thymine is then uracil. Thymine, then uracil. Huh? I didn't get you. Sorry, Jia. Okay, perfect. Let's go ahead with the next question. Select the correct match. Australian marsupials are given over here. Placental mammals are given over here. You have to tell me which is the correct match. Sudarshan says the answer for this is D. By the way, everyone who gave me the correct answer previously, Yashwant, Manya is there. Hello, Manya. Vinay, I remember you, Manya. And Vinay, if at all you are the same person. And Malika, Mansi, Dhanush, everyone. Prithvi. Perfect. If at all I am forgetting to take out your name, please forgive me. Prachi also, Shreya also, Paru also, Jia also. Everyone says Rakshita also. Sorry, Rakshit also. Shri Rak. Okay, fine. Everyone says the answer for this question is what? D. Someone has said it is A and all. So D is what? Spotted cuscus and lemur. Is this answer correct? Listen to me very carefully. This particular thing is what do you call? Based on adaptive radiation. So it was found that there were a group of organisms that is known as Australian marsupials. Baba. Australian marsupials. So these were the ancient organisms which are there. Some of them went this side, 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 some of them went this side. What do you call this as? Basically, they moved. They went different direction. This is radiation, and they basically got adapted there, which is why this is an example of what? Adaptive radiation. And if at all you have seen something over here, they basically had a common ancestor and they diverged, which is why this is an example of what? Divergent evolution. Very, very important thing that I am explaining over here. What is this an example of? Divergent evolution, because from a common point they are diverging, right? Now what if I tell you that one more group of organisms came into the picture and the name of those organisms were placental mammals. So Australian marsupials were different, placental mammals were different. They also had a common point over here. Some went over here, some went over here, some went over here, some went over here, some went over here. What are they showing? They are also showing adaptive gradation. What are they individually showing? They are also showing divergent evolution. From a common point, they are also moving. But you need to understand one very important thing. When I talk about both of these organisms together, they were from different ancestors. They came to a point. They are inhabiting in the same habitat. Obviously, if at all they are inhabiting in the same habitat, they will have some similar feature, right? So can I say, now when we talk about two organisms together, it can be an example of what? Convergent evolution. So individually they were showing divergent evolution, but now they have converged to a point, they are showing convergent evolution. So your question is, if from the Australian marsupial side, spotted cuscus came, okay, in the same group, my dear children, which organism came from placental mammal side? It was lemur. It was lemur. So your answer for this question is D. And here I'll be giving you a trick to remember, and that is, listen, in your NCERT you have seen this particular picture. I hope you have seen this. This is in evolution chapter, right? Placental mammals are given over here, Australian marsupials are given over here. You have mole here, then you have marsupial mole here. If at all you see the names, the shorter names are belonging to placental mammals and the larger names are belonging to Australian marsupials, right? So Australian marsupials itself is wrong, so you can remember that it has longer names. So mole and marsupial mole, remember, it is very easy to remember. Anteater and numbat. This is something that might be confusing, so please remember. Anteater from this side and numbat from that side. Okay, mouse, marsupial, most easy to remember. Lemur, spotted cuscus, difficult. Again, remember, the longer name is on this particular side, and here you have lemur. Okay, flying squirrel, flying phalanger. Flying squirrel is here, flying phalanger is here. Bigger name is still over here. Now, bobcat, Tasmanian cat, okay, and what do you have? Tiger cat. Here you have wolf, you have a Tasmanian wolf. So please remember, you can get other things also. Instead of spotted cuscus and lemur, you can get anteater and numbat, or you can get probably flying squirrel and flying phalanger. Know your answer. That is very, very important. Okay? Okay? And yes, Manya, first uracil and then thymine. Uracil came first in RNA. Listen, RNA was the first genetic material. It was highly unstable, which is why you had got DNA. Okay? Ma'am, my name is Chirant. Dear, I'll try very much to remember it. Sometimes I might not say the name. Please forgive me, okay? Ma'am, I'm scared of 5-marker hot question. Do not worry, I have covered it. The video length is 4 hours 48 minutes. I recorded it. Okay, so just a second, okay? Yes. Yeah, so please make sure you watch it. I will make sure that it is released for you guys as well. Okay, perfect. So those of you who gave me the answer as D, perfect. Now you have one more question. Which of these ancient men protected their body with hide and buried their dead? Can you try this particular thing? Ma'am, how many hours would you take, ma'am? Lakshmi, I think it might go, so other teachers I asked, they said that it took around 5 to 6 hours. So I'm also expecting the same. If at all you guys are quick and if at all I do not see much doubts, it will be fast. Sudarshan says the answer for this is B. Lakshmi says it is B. Mansi says it is B. Rakshit also B. Prachi B. Praru B. Prithvi B. Manya B. Okay, Kids Zone. Malika. Perfect. Harshika, Neanderthal man. Yes. Shrirak, everyone are saying, Vinay Shetty also saying BB. Ma'am will call Baba instead of your name. Exactly. So the answer for this question is Neanderthal. Always remember, my dear children, Neanderthal organisms had the highest brain capacity. It was 1400 cc. Okay, not even us. We even do not have 1400 cc brain capacity, but they had, right? And basically they were the first ones to protect their body and also bury their dead. They had some emotions ongoing in them. And that's pretty cute, right? Cute. So your answer for this question is what? B. Does anyone want a proper chart as of now itself, or are you okay if I just go on teaching? If it's fine, I will just go ahead, but if at all you want me to tell which organism came first, I can do that right now. Sri Raksha, I'm so sorry, Sri Raksha. So everyone go on, fine. So I'll be going on. By the way, guys, I have put over here, you can read it again. If at all you have watched my videos, you will be able to get it as well. Hi ma'am. Yes, someone was asking my name. My name is Jasmine. Okay, my name is Jasmine. Yes. So human immunodeficiency virus replicates in. Human immunodeficiency virus is what, Baba? HIV. It is HIV. Ma'am, first thymine came or uracil? Baba, uracil. It is RNA that came first, right? RNA was unstable, which is why you got DNA. Fine. So uracil was the first thing. In RNA you had uracil, and then in DNA you got thymine. Fine. Okay. So my God, you guys are super fast. The answer for this question is C, is what you guys are saying. HIV virus replicates in B cells, that is B lymphocytes, natural killer cells, polymorphonuclear leukocytes, or basically helper T cells. The answer for this question is helper T cells. Always remember, HIV virus which is there, first will be going and attaching what? Attacking macrophages. What? Macrophages. These macrophages are known as the HIV factory. Why do I call it HIV factory? Because this is where they will divide and increase their number. But you know what? Their actual enemy, okay, or their actual target is what? T helper cells. T helper cells. Why? Because this guy is the papa guy who is helping around building our immunity. If at all I target this particular guy, don't you think our immune system will go weak? And that is exactly the name of this organism, human immunodeficiency virus. It wants to make us immunodeficient. So always remember, actually it is macrophages, which is HIV factory, but the target is T helper cells. And since you do not have macrophages in the option, you can go for T helper cells. Okay? Ma'am, first in macrophages, then in T cells. Exactly. Okay? So in T cells also they will replicate, they will do all the things out there. But yeah, initially it is this, the HIV factory. Perfect. Now let's go ahead. By the way, guys, have you subscribed to the channel yet? All of you who are newcomers, who do not know about this channel, have you subscribed to the channel? Yes? Please go watch it. Ma'am, you are amine with the A. Oh my God. Nice pickup line. Okay, 9. The drug which is depressant and slows down body function is? The drug which is depressant and slows down body function is? Okay, so many people are saying A. Sudarshan A, Prachi A, okay, Manya A, Mansi, Prithvi, and Hassan. Hassan, hello. Okay, everyone. Aisha is also there. Kid Zone is also there. So your answer for this question is A, smack. Why do you call it smack? First of all, there are two things which makes your body, what do you call, go into the sleepy phase. One is you have morphine and the other one is heroin. Okay, both of them come under opioids. They come under what? Opioids. Morphine is a sedative. That means it makes you sleep. Apart from that, it's a painkiller. So a doctor who has done a surgery probably gives it to a patient who cannot bear their pain, because this acts as a painkiller. That does not mean if at all we have pain, we should be taking it up. No, we should not be, because high doses can also kill someone. Now, what about heroin? Heroin is someone that is going to slow down your body, and also it is going to act like a depressant. And the other name for heroin is smack. The other name for heroin is what? Smack. Apart from smack, it also has one more name, and that is known as ayoyo. And that is known as diacetylmorphine. What do you call it? Diacetylmorphine. Why diacetylmorphine? Because this particular guy, morphine, underwent a process that is known as acetylation to bring heroin. And that is why it is known as diacetylmorphine. Please know all the things around this particular thing. Okay, it's not like, okay, smack. That's the only thing I have to remember. No, you should know each and everything about this particular, drug out there. That is how the questions are going to be framed. Okay, so those of you who are asking me, will the exact question come from the model question paper? No, concepts are going to come. So please know about it. That is super important. Okay? Yes. Whew. Fine. So your answer for this question is A. I have given you the name of the whole thing. Make sure. Crack and coke. Crack and coke is cocaine. CC, no? Crack and coke. Both of them are cocaine. Fine. Let's go ahead with the next question. Saccharomyces cerevisiae, commonly called as brewer's yeast, is used for fermenting malted what? Fermenting malted what? Wait a second, some questions have come in. Ma'am, I'm worried about biology 2-3 marker question. Asya, my dear child, I have done the whole video on it. Why are you worried? Go watch it. Okay, go watch it, and if you have doubts, put it down in the comment section under that video. I'll take it up. Ma'am, do morning revision class. Definitely, we'll be doing that. Sri Raksha is asking PMNL function. PMNL is polymorphonuclear leukocyte. Polymorpho means this nucleus has no multiple morphology. It looks like multiple lobes are there in the nucleus, right? And it is also known as neutrophils. What do you call it? Neutrophil. That is the easier name to remember. Now this neutrophils are the ones that does phagocytosis, as in basically suppose some pathogen is there, it will take it in and kill it. So neutrophils, macrophages, they are all phagocytes. They do the similar function. But if at all something comes first, the first guy to come in will be neutrophil. Then comes macrophages and things like that. Okay? Fine. Okay. B is what everyone of you guys are saying. What is B? B is what? Cereals. Cereals. Just a second. So Saccharomyces cerevisiae, commonly called brewer's yeast, is used for fermenting malted cereals. Is that the answer? Listen, what do you mean by malted? So suppose there is a grain over here, there is a cereal over here. This cereal was about to germinate. You gave all the conditions such that it was about to germinate, and then you took it away. So basically it is in the stage where it is about to germinate, and then you are doing fermentation. Why? Because when something is about to germinate, it will be using up the starch and will be starting to break it down. Now, when it starts to break it down to the glucose, basically it will be easy for the fermentation process to happen, because what happens in fermentation, the glucose breaks down to pyruvate, the pyruvate breaks down to ethanol, so you needed glucose, right? And the easy way to get that is by using malted cereals. Also remember, it's not just malted cereals, and also there is fruit juices. Malted cereals and also fruit juices are something that you use for fermenting things, okay? Or for production of things out there. Yes. Question number 10. In the year 1963, the two enzymes responsible for restricting the growth of bacteriophage in E. coli were isolated. One of them cut the DNA and the other added the group. Listen, in 1963, an experiment was being done. A thing was being found. That is, in E. coli, inside the E. coli, there were two enzymes being found. One enzyme was the one that used to cut the DNA. Other enzyme was the one that was doing something. What is that enzyme and what is it adding? That is the question. Not what is that enzyme, what is it adding? That is the question. And by the way, every one of you who are giving me the answer, I am seeing you, okay? Naja, Aisha, the Deepti, Hassan, Prithvi, and Lakshmi, Sri Raksha, Jia, Malika, Hassan, everyone. Riaz, hello dear, hello. Rakshita, Adnan King, hello. Okay. Mania, perfect. Okay, ma'am, your explanation is just like wow. Thank you, guys. And Kids Zone and everyone is saying the answer is A. Okay, A is what? Methyl. So again, I'll tell you, listen to me very carefully. E. coli is there, it has its own genetic material, right? Suppose a bacteriophage came. Bacteriophage is what? A virus. Bacteriophage means it eats up bacteria, that is why it is known as bacteriophage. So bacteriophage came in and put its DNA over here, basically genetic material over here. What is going to happen? The whole ultimate target is to basically get incorporated into here. All the viruses like this only. They want to get incorporate and use the machinery. But E. coli, my dear children, developed two enzymes. One enzyme is basically nothing but the restriction enzyme out there. What does the restriction enzyme do? Go and cut the DNA. Now listen to me. If it can go cut that particular genetic material, it can also cut its own genetic material. So then E. coli thought, I have to do something so that I can protect my genetic material, which is exactly why it again produced an enzyme, and this particular enzyme did one very important thing. It added methyl group around. Okay, it added what? Methyl group. Now when methyl group is added, it's like a signal that you should not be touching me. Okay, you should not be cutting me. So basically the next thing that was added was methyl. And please remember, even though this was found, the first restriction enzyme which was there, it was not EcoR1 or things like that, it was HIND2. The first restriction enzyme to be isolated and discovered was HIND2, even though all this was found before that, but they could not name this particular thing. They just knew something like this was happening. Okay, ma'am, autosomal disorder, we'll come to that, dear. As of now, we'll go question by question. Listen, I'll take a separate video on what can be done, like if at all you have any doubts, what should we do about that. But as of now, we should not be, okay, because otherwise this particular lecture is going to go on, and so many children are joined to get here for solving the model question paper, right? So that is the reason. Ma'am, can we follow the blueprint to study? Because I saw past it was not according. Winter Luna, so it's not exact, it might not be exactly according to the blueprint, it can be according to the blueprint as well. For example, in biology, the previous year, it was not exactly the same, but for the other subject, it was. Okay, for some subject, it was. Okay. Perfect? Yes. This is the first MQP, dear. This is the first MQP. Okay. Now, question number 11. Which of the following is the correct representation of the primer annealing step in PCR? Okay, which one is the first correct representation of primer annealing step in PCR? Primer annealing, you know. So, to make a new strand, always DNA polymerase, we need a ready-made primer. So, you are being asked which one depicts that one correctly. So, okay, C, Rakshita says C, Paru D, okay, Aisha C, user Anbu is there, C, okay, C, A, B, C, D, you have given me all the multiple choice options to there. It's okay if at all you ask doubts, okay, all the children who are asking doubts, I truly, it means a lot of, what do you call, I understand that you're relying on me for doubts, and that's really great. Okay, so do not be sad if at all you're asking doubts. The only thing is that I'll take it afterwards. I'll make a separate video probably for taking up your doubts, yeah? Yeah. So since you guys are asking so many things, let me see what is the actual answer for this particular question. The actual answer for this question will be D. Will be D. Okay, it is going to be D. Why D? So listen to me. This is in 3' to 5' direction. In the new DNA strand will always be in 5' to 3' direction, right? So new DNA strand will be going from here to here. For that you need a primer up over here in the 3' end, right? So that is happening over here. In the same case, at the 3' end you have a primer. And the 3' end you have a primer being attached such that a new strand can be formed in 5' to 3' direction, right? Which is why your answer will be this. Why it is wrong? Over here it is correct, but over here it is in the 5' end. So in the 5' end, if at all a primer is attached, then the new strand is going to be formed in 3' to 5' direction. Is that the rule of DNA polymerase? No, Baba. Here also it is in 3', but here it is in 5'. Again wrong. Here it is in 5'. Here it is in 5'. Again wrong. Always remember it has to attach at the 5' end only. Okay, so your answer for this question would be, sorry, at the 3' end only, not 5' end, 3' end only. So which is why your answer for this question will be D. Okay, cool, cool, perfect. Next one, question number 12. Given below the steps in the gene therapy of a patient with adenosine deaminase deficiency. Now this question, make sure you read it properly. Okay, read it properly and then only go ahead. So let's see. Lymphocytes are grown in a culture outside the human body. Okay, lymphocytes are altered with the functional ADA gene. Okay, lymphocytes are collected from the blood of the patient. The genetically modified lymphocytes are infused back into the patient. You have to go step by step and tell me which one is the correct order for the steps that you use for gene therapy. Okay. Lakshmi says, Lakshmi had a different name, I remember. Ishta says it is C, and Rakshita also says it is C, and Sanjana C, Paru C, Prachi C, Mansi, Deepti, okay, Anbu Gowda, Jia, and Prithvi, Shreya, everyone, Sudarshan, everyone says it is C. Is it C? Let me see. C is what? 3124. Okay, 3124. Let me go according to that. So lymphocytes are collected from the blood of the patient. Yes, that has to be the first one. So do we see any three options out there? No. So we cut out these options. We keep this and this. Fine. Now you have to look from the options and then it saves a lot of your time. Okay. So after 3 it is 4 over here. What is 4? The genetically modified lymphocytes are infused back into the patient. We have just collected it. How can we infuse it back? So again, this will be wrong. You are basically wasting less of your time out there. Now, if at all you see this option, after 3 you have 1. Lymphocytes are grown in culture outside the human body. This is a point that we usually miss. Listen to me. Lymphocytes are taken, then we culture it outside the body. This point has to be taken care of. After culturing it only, you are going to alter it with what? The functional ADA gene. After putting ADA gene into the lymphocytes, you are going to put it back inside the body, which is why your answer for this question is going to be C. Okay, your answer is going to be C. For those of you who got it wrong, okay, fine. Let's go ahead. Question number 13. The organism that reproduce only once in their lifetime. The organism that reproduce only once in their lifetime is what? Ma'am, you are the best. The other part I will not read. Okay. So C is what? Salmon fish. Salmon fish is the one that reproduces only once. Is that true, guys? Is that true, guys? Yes, the answer is correct. Along with salmon fish, is there any other example that is given in your NCERT that you need to remember? Think. Everyone who is watching this video, think extra over here when you solve a question. Is there any other organism that breeds only once in their lifetime apart from, yes, the Deepti says it is bamboo. Hassan also says it is bamboo. Mansi also says it is bamboo. Yes, it is bamboo. Please remember, salmon, fish and bamboo, they breed only once. And what do you call these organisms as, which breed only once? You call them as simulparous organisms. Simulparous organisms. And the ones who breed, multiple times in a generation are us, mammals, and also birds. You call them heteroparous organism. Apart from that, in the same paragraph, one more thing is being given, and that is what, let me think. One more thing is given, yeah. Some organisms are there, they produce so many progenies all at once. Why? Because they know that the survival rate of these progenies are less. Only few will survive. Example for that is pelagic fishes. Pelagic fishes. Okay, pelagic fish means basically they are not inside the water, they are not towards the shore, they are in the middle, in the open sea. Those are pelagic fishes. Pelagic fish is an example for the ones that will be breeding, that breeds so many progenies all at once. Okay, so please remember that also. Okay, ma'am, are we belonging to yeah dear, we are mammals. Okay, we are mammals. Yes, perfect. So I have written over here, some produce a large number of small-sized offspring. For example, oysters, pelagic fishes. Know the examples. Okay, oysters and pelagic fishes. While other produce a small number of large-sized offspring. For example, us. We produce small number, only 1 and 2 children out there in every household, right? I just have a brother. And now, basically, because we know the survival rate is more in comparison to these organisms. Okay, cool. Now, question number 14th. Let's talk about this and then I'll take the routes. The decomposers especially abundant in the bottom of the pond are. The decomposers which are there in the pond are what? Ma'am, are there one-shots of every single chapter? Yes. So I think biodiversity and conservation is not there. Just every chapter is there in Master Stroke series. Go to playlist section of the channel and then you will find Master Stroke series. In that it will be there. Okay? Okay. Bacteria in, okay. Someone is saying D, A, B, C. Guys, what is the answer for this question? D is what do you say? D is what? Flagellates, fungi and bacteria. The answer for this question is this. And right now, those of you who makes the teaching very, very bad, I will be putting them on timeout. Okay. So bye, King. We'll see you really, really soon. Okay. So all my dear children are right here. D and C. D and C. D is the correct answer. Listen to me. So usual decomposers which are there, they are FB. FB means what? Facebook. Facebook. Yeah, Facebook. So you can remember it like this. It is fungi and bacteria. It is fungi and bacteria. But in the case of ponds, you have an extra thing that is there, and that is flagellates. And that is flagellates. Do not forget it. They also act like decomposers over here. Okay? So your answer for this question is going to be D. Works? Works, ma'am. Perfect. Now, 15th question. The IUCN Red List documented the extinction of species in the last 500 years. Include what? Include what? Adnan, tell me, dear. Some say that they love your voice. Oh, thank you so much. That's so sweet of you. Okay. Now B, Hassan and Anbu Gowda and Mansi, Sanjana, Prachi, Prithvi, Lakshmi, Disharam and Krik and Cypher Sports. I forgot the name that you told me. But yes, it's Janu. Okay, perfect. Everyone is saying B. What is B? 784. 784. What is the 784, Baba? This is the documented number of extinct species which are there. And in this 784, you should know how many vertebrates are there, or basically how many invertebrates are there, how many plants are there. This is very, very important. Yes, Swaroop, exactly. So you should be knowing about the number. Can anyone tell me the number of that also? I have discussed this in all one marker. I expect you guys, at least few of you guys, to remember it. Can you try to remember? When we say 784, I'll give you a hint. 87 of them are going to be plants. 87 of them are going to be plants. What about others? Okay, guys. (369) invertebrates, okay? I want all the things. Okay, vertebrates is this. Invertebrates is this. 87 plants. Guys. You're keeping on guessing game. Give me one answer, all of you. Vertebrates how many? Invertebrates how many? This one I gave. That is 87 plants are the one which is going to be there. Apart from that, this number is important. You may get questions on the basis of this also. Do not forget it. I'm telling you, model question paper is for you to understand which is important. Any idea? Since you guys are not saying, let me see. It is 338. Okay, please do not forget it. It is super important. 338 vertebrates, exactly, Mansi and Hassan also. 338 vertebrates, 389 invertebrates. No, Baba. Listen, these are the numbers. 338 vertebrates, 359 invertebrates, and 87 plants. This is the correct number that you have to remember. So when we say 784 species are extinct, please remember this number. The highest number is what? It is invertebrates. It was 359. In invertebrates, it was 359. Then comes vertebrates, 338. Then comes birds, plants out there, which is 87. Okay? Fine? Perfect? Cool. By the way, did you understand it? So answer is right over here. You can get this also. If you understood it, can you just give me a quick thumbs up, children? If you understood this, can you just quickly give me a thumbs up? Because by this we are ending all the MCQs. 15 MCQs of model question paper 1 is officially done. Thank you so much, Vinayak. You guys are also so cute. And thank you all of you for giving me the thumbs up. Okay, let's go ahead. Ma'am, I got Kantara Beach. Perfect. Great. Okay. Let's go ahead. Before going ahead, one more very important thing, that is we have officially launched the KC30 batch. So anyone who is interested for a 30-day crash course program after your boards, you can take it up. The pricing for this is rupees 2499. The offer price is there. It is ending at 25th of February. What do you get? You get complete syllabus coverage. There will be chapter-wise KCET PYQs also. And this chapter-wise KCET PYQs, along with giving you solutions and things like that, there will be the test conducted in what you call a proper format with timings and all. And there will be 10 full-length mock tests. All of it is going to start from 21st of March. So if at all you're interested, go enroll for it. Yeah? Yes. And over here, let's go ahead with solving one question. So I want one question. Okay, 16 question 1A, whatever is the answer, you can tell me. 17 questions. So we'll go one by one. Fine? So, yes, Jia, as for what I remember, it is that. It is that only. Okay? So the initial few days of milk produced after pasteurization is, so once the milk is being produced, what is the first initial few days milk known as? Colostrum. Sheikh Mohit said colostrum. Hassan also said colostrum. Prachi also said colostrum. Yes, Baba. So your answer for the 16th question is what? Colostrum. And colostrum is rich in which antibody? It is rich in which antibody? It is IgA, immunoglobulin A. So you can remember A for Amma. From Amma only the first milk is there, right? So A for Amma, IgA. Remember that. Okay? Guys, we'll go one by one only. Okay, we will go one by one. And next in, question number 17. Oral pills are contraceptive, effectively functions by inhibiting what? Oral pills which are there, which act as contraceptives, my dear children. What does it affect? Amoebiasis, richness, nostoc, ovulation and diversity is given over here. Answer is ovulation. Listen. Estrogen and progesterone are hormones which are super important for the ovulation process to happen. Especially estrogen is there, right? In oral pills, what you're going to give is that you're giving additional estrogen out there. And when you're giving additional estrogen, what is going to happen, Baba? The balance is going to be shaken up. When the balance is going to shape up, it would not do the similar work that it used to do. So ovulation is not going to happen. Implantation is also not going to happen. So ovulation is something that is being inhibited. So your 17th answer is this. So everyone who said ovulation, perfect. Okay, so some people went to 18th question also. Okay, fine. 18th question. Symptoms of constipation, abdominal pain and cramps is associated with which disease? Constipation is there, abdominal pain is there, cramp word is also being used. Whenever cramp term is used, just understand it is amoebiasis. It is amoebiasis. It is also known as what? Amoebic dysentery. Okay, it is also known as amoebic dysentery. Why amoebic dysentery? Because over here in the stool, what do you call, blood will also be there. Blood and mucus is also going to be there in the stool, which is why you call this as amoebic dysentery or amoebiasis. So for 18th question, your answer would be what? Amoebiasis. Oh my God, Lakshmi Ravi gave me the answer as a whole. Okay, next thing. 19th, cyanobacteria is an autotrophic microbe widely distributed in aquatic and terrestrial habitats, can fix atmospheric nitrogen, is what? So you have to give the example of cyanobacteria. Cyanobacteria. So cyanobacteria is nostoc and anabaena. Nostoc and anabaena, and also oscillatoria. So you know there is a game Uno. There is a game Uno. So similarly over here you can remember Ano. So A for anabaena, N for nostoc, and O for oscillatoria. These are bacteria, but they are cyanobacteria. What is the difference between bacteria and a cyanobacteria? Cyanobacteria mostly are organisms that can prepare their own food. They can prepare their own food. Okay, always remember they are autotrophic in nature. And the example for that is this: they can also fix nitrogen, and nostoc is given over here, so which is why your answer for this question would be 19th. Okay, fine. Next, going ahead, Alexander van Humboldt observed that within a region, species dash increased with increasing explored area, but up to a certain limit. So listen to me carefully. Richness. Parvati says richness. Prachi also says richness. Mansi also says richness. Okay, listen to me. Yes, Hassan, you also call them blue-green algae. Cyanobacteria is also known as blue-green algae for the same reason. That is, they do photosynthesis. Sorry, they do autotropism. Okay. Richness. So Alexander van Humboldt, he said that basically tomorrow if I move out of this area, I'll be seeing more species. If I move even more outer, I'll be seeing more species. What do you understand from that? Basically, when the explored area goes ahead, the species richness is also going ahead. So that is the thing, but only it happens only up to a certain limit. It's not like I will be seeing n number of species. There will be a finite quantity to it. Okay, so over here your answer for this question would be richness. It will be richness. So where is richness? Richness is over here. It is not species diversity, it is species richness. Do not forget it. Alexander ends with an R, and R stands for richness. If at all you want to remember that, you can remember it. Okay? Okay. Perfect. Oh my God, there are so many comments. Okay, just a second. So now we enter into the next phase, and that is the 2 marker questions. Before that, let me just read your things. Oh my God. First of all, guys, please do not spam. Do not spam. Okay. Second thing, Vishnu is asking, ma'am, we will be in the crash course because we love your teaching. Thank you so much. Yes, I'll be teaching you. And how lovely. Thank you, Sohail. And everyone who gave me the answers until now. Everyone, I want to read out their name. Anbu Gowda, Paru, Prachi, Mansi, and Adnan King, Vinayak, Deepti. Deepti, you were asking some question of some page number. I could not recollect the comment, and you can put it back. Jia, Disharam, everyone. Okay, if at all I'm forgetting someone's name, please forgive me. All of you are doing good. Ma'am, what about 2 and 3 marker? 2 and 3 marker of model question paper are going to be discussed over here. Apart from that, I have made a separate video on it. Please make sure you know that. Yeah, please make sure you watch it. Now, just a minute. Okay, for spots, dear, I do not know the answer for that. So I know there's a feature like that in YouTube, but I really do not know that. Yeah? Okay. Guys, come back to the question. So draw a label diagram of mature 2-celled pollen grain. How do you draw it? Listen, over here, this particular question is important because I saw certain things about it. If at all you know, but listen to me carefully. There is a very big cell. You call this as what? Vegetative cell. Vegetative cell. You can also remember very big cell. Okay, V for very big cell. Then you have generative cell. Generative cell. Why generative cell? Because it is going to generate something. What is it going to generate? Gametes. How many gametes, Baba? It is going to form 2 gametes at the end. Fine. Now the shape of the nucleus. This is very, very important. What is the shape of the generative cell's nucleus? It is irregularly shaped. Okay, this question came, I think, previous year, the model question paper had this question. Irregularly shaped nucleus is there, whereas over here you have an elongated nucleus. You have an elongated nucleus. How did you get 2 cells over here? How did you get 2 cells over here? The reason is that there was mitosis. So from a microspore, you got pollen grain, right? So the microspores which are there, they undergo mitosis, and that is how you get a very big cell and a small cell. This particular layer which is there, it is known as plasma membrane. What do you call this particular layer as? This is known as your plasma membrane. But apart from it, does it have any other layers? The answer is yes. It has a continuous layer which is like this. This continuous layer which is there, you call this as an intine. Okay, intine. And intine basically is the first thing that that is made up of Priyanka Chopra. What do you mean by Priyanka Chopra? P for pectin and C for cellulose. So intine which is inside is made up of pectin and cellulose. Okay, ma'am, outside it, does it have another layer? The answer is yes. This layer is discontinuous. This layer is discontinuous. What do I mean by that? Basically, there are pores over here, right? Okay, ma'am, what is this layer known as? This is known as exine. What is it made up of? Sporopollenin. It is made up of sporopollenin. It is the most toughest organic resistant material out there. You have to remember about sporopollenin. This is the most toughest material out there, and basically it can resist alkali, acids, any pH out there, which is why the pollen grains can be stored as fossils also. Why? Because it is exine is made up of sporopollenin. Fine. And whenever there is no exine, there is no sporopollen over here, you say that this is a germ pore. Okay. Now, what is the use of a germ pore? Listen, through it only the pollen tube is going to be formed. Okay, how is the pollen tube formed? Listen, so this is your intine. This intine goes out like this, making the layers of the pollen tube, right? Now, what is there inside the pollen tube? Inside the pollen tube, the cytoplasm is there of the vegetative cell, which is why we call the vegetative cell also as tube cell. You also call vegetative cell as what? Tube cell, because it forms the inner thing. Now, inside the tube, or basically pollen tube, what do you have? You have the male gametes. So usually it can be what do you call a generative cell which is released over there. Later, suppose when the pollen grain lands on the stigma, this generative cell can divide into two, giving rise to two male gametes. This happens in 60% of angiosperms, as in when the pollen grains land on the stigma, that is when division happens to form the two male gametes. In 40%, the rest of the angiosperms, it is already divided into two gametes out there, and that is how it is going to land. So please know that also. Okay, got it? Okay, now I'll read your things. Why are you ignoring? Whom am I ignoring? Listen, when someone teaches you, okay, when someone teaches you, suppose you are teaching me, can you read the comments out there? No, you will be focused on teaching. Why are you teaching? Because you want the children who are watching to score good, right? So please do not take it otherwise. When I am teaching you, I am teaching you. Okay, when I give you a time, you can ask me your questions. Okay. Sohail, it can, as in 5 marker, you have a whole different thing happening. I'll be showing you the 5 marker questions which usually come in. Okay? Yes. So, children, is this particular question clear? Vijaya, yes, vegetative and generative, perfect. Can I go ahead, children? Yes. Next thing. Draw a label diagram of a mature two-celled pollen grain. So, see, this question is this how you have to draw it. Make sure you draw it properly. And now you have mentioned any 4 assisted reproductive techniques that can be advised to couples with infertility problems. Listen, you are talking about assisted reproductive techniques, and you are talking about couples with infertility problems. So it can be a problem with male, it can be a problem with female. But the first step that you think about is IVF. What is IVF? In vitro fertilization. That means fertilization outside the body, that is in the laboratory condition. For that, what do you need? You need a sperm and you need an egg. Now you can take the sperm and egg from the parent itself who are not able to conceive, right? Suppose they have all the other features, that is the mother is having a proper uterus and everything, then you just need to take the egg and the sperm. You need to do the fertilization outside if fertilization not happening inside, right? And then you get a zygote. Then you get what? A zygote. Zygote is a single cell thing. This zygote, or basically the zygote divides and form 8-celled embryo. Up to 8-celled embryo, you can transfer it where, Baba? You can transfer it inside the fallopian tube. This is known as ZIFT, zygote intrafallopian transfer. So zygote up to 8 blastomeres is transferred into what? Into the fallopian tube. This is super, super important. But only up to 8 blastomeres, remember that. Okay, ma'am, what if the embryo is more than 8 blastomeres? Where do we transfer it? Can we transfer it in the fallopian tube? No. If at all you transfer it in the fallopian tube, it will lead to ectopic pregnancy. That means pregnancy in the fallopian tube. It can lead to death of the mother as well as the child, which is why you need to do it. You need to do intrauterine transfer. That means inside the uterus. Intra means inside. Uterine means uterus, right? Intrauterine transfer, where embryo with more than 8 blastomeres can be put inside the uterus. Fine. Now suppose there's another technique. Suppose a female is there, she has all the what do you call features of basically bringing up a child in the womb. Her uterus is perfectly fine, but the gametes are not being formed. Probably she has a rudimentary ovary, she has a problem with some ovarian problems out there. In that case, what do? Basically, you can do gift. Gift is what? Gamete intrafallopian transfer. You take the gamete from someone else, and basically the donor gamete is put inside the fallopian tube. Why again inside the fallopian tube? Because fallopian tube is a place where the sperm will come and fertilize. That is the reason you put it inside the fallopian tube. So that is known as gift. And always remember over here, when someone performs gift, gamete intrafallopian transfer, fertilization is going to happen inside the body, not outside, because you have transferred the gametes, right? Now you're just waiting for the sperm to come. So fertilization is going to happen inside. What do you call this fertilization as? In vivo. It is not known as in vitro, it is known as in vivo. Done. Now last one that we have is artificial insemination. What do you mean by artificial insemination? Basically, suppose a man is there. Suppose a man is there who is having erection problem. Okay, the penis is not being erected. In that particular case, can he transfer the sperm? No, that is a problem. Also, if at all there is a man who does not produce enough quantity of sperm, or the quality of the sperm is deteriorated, in that case, what do you need? You need the sperm from some other donor. Okay, or probably if at all there is only erection problem, you can have the sperm from yourself, and then the doctor is going to put this particular semen inside the reproductive tract. That is known as artificial insemination. It is known as artificial insemination. Semen from the husband or the donor is artificially introduced in the female reproductive tract. If at all it is done directly inside the uterus, you call this intrauterine insemination, IUI. So IUI is a type of artificial insemination. When you're doing it exactly inside the uterus, you call it IUI. I hope this question is clear. These are the 4 assisted reproductive techniques that you can write. Preetham, it is live, dear. Dhanush, ma'am, I still need to complete the previous lectures. You can. Okay, you can. You just find some time for that and sit with the questions as such. Okay, you will be able to do it. Perfect. Thank you guys for giving me the thumbs up. Thanks for the great explanation. Thank you, thank you, thank you. Ma'am, important diagrams, I will be making a separate video on that. But whatever diagrams have been completed until now in the all 1 marker, 2 marker, 3 marker and all, that is the most important ones. Yeah. But yeah, I'll be making a separate video also. Fine? Gia? Yeah. Next question. This is important. This is confusing also for some people. Question number 23. Recombinants obtained in both Mendelian and Morgan experiment, yet the process involved are different. Comment. So basically in Mendel's experiment, if at all you have seen, when you get 9:3:3:1 ratio, right? And do we need to explain this zift and gift and all? Exactly, you need to. Okay. Little bit, one liner could be enough if a 2 marker question is coming. So in the case of Mendel's dihybrid cross, we studied that in the first generation, they got something like this, right? And they did selfing. They did selfing. When they did selfing, they noticed one very important, that is, you have chromosomes for capital R, small r. You have chromosome for capital Y, small I. Now, when you make gametes for this particular plant, you need to understand that from this pair of alleles, only one guy will come in, right? Only half the number of chromosomes is going to go in the gamete. Similarly, from this also, only one guy will be coming in. Suppose R and capital Y came in. In the next gamete, capital R and small y can also come in, right? That can happen. In here, small r can come from this side and capital Y can come from this side. In here, you can get small r and small y also. So what do you understand from that? Alleles of one particular character, that is capital R, small r, is going to assort independently on the alleles of other pair of things. So basically capital R does not say that I will only go with capital Y. There is nothing like that. Okay, it can go with capital Y, it can go with small y. This pair of allele segregation has nothing to do with the next pair out there, which is why you get multiple type of gametes, which is why you get multiple type of organisms also at the end. You started with round and yellow and wrinkled and green, but then you got round and green and wrinkled and yellow. New combination out there, non-parental character. Why? Because of independent assortment of gene. So in Mendel's cross, recombination occurs due to independent assortment of genes on different chromosome. But when you talk about Morgan's experiment, why do you think there was recombination happening? Morgan spoke about crossing over. So listen to me carefully. Over here, suppose I have A gene and Small, capital A here, small a over here. Capital B over here, small b over here, right? Now suppose, since the chromosomes are very aligned to each other, they are very close to each other, what is going to happen? There is going to be exchange of some parts of the chromosome. When this exchange happens, what is going to happen? You'll get a new type of chromosome. What is that new type of chromosome? Capital A will still be here, but here you will be having small b. Similarly, you will have small a over here, but here you have capital B. Why? Because this got exchanged. Now, don't you think there's a new combination of chromosomes being formed? Yes, Baba. And now this new combination gives rise to what? New type of organisms out there. That is why we look different from our parents. Even though they are giving their chromosomes to us, we look different. Why? Because there is recombination happening. There is crossing over between the linked genes. So these are the genes which are there, and there is crossing over that is happening. That is the reason we get recombinants. Is this particular thing clear to all of you? This is super important. Make sure you write the answer properly. Neeraj, ma'am, do full marathon of all the chapters live. We'll be definitely doing it towards the examination, okay? Hi, ma'am. Hi, Bhanu Prakash. Children, is this clear? If it is clear, can you just give me a thumbs up? Ma'am, how can I join with you in private? Oh my God, that is literally difficult for us as of now, Lakshmi. I wish I could give you an answer for that. Okay. Yes, clear. Thank you so much. Perfect. The next question that we have is question number 24. Okay, let's go. Question number 24 is, name the methodologies used in human genome sequencing. So human genome, you will get a question on human genome sequencing for sure. So you can go study about the aim of human genome project. You can see the organisms which were used in human genome project apart from human beings. There was C. elegans, there was bacteria, there is rice, Arabidopsis. All of this was also being sequenced. So please know about this. Okay. So now we come over here. What was the technique used for human genome sequencing? First is expressed sequence tags. What do you mean by expressed sequence tags? Basically, only the genes expressed as RNA were identified. So remember over here, suppose this is your DNA is very long. Only some part of it is going to be undergoing transcription. Okay, it is going to make RNA. Some part is not going to undergo transcription. So basically the one that undergoes transcription and forming the RNA, those sequences were only identified. That is what you mean by expressed sequence tags. That is what you mean by expressed sequence tag. Exactly, MP Myler. Perfect. Hi. Okay, now next thing, whole genome sequencing is also there. That means whether it is coding, whether it is non-coding region, each and every part of the genome was being sequenced. That is whole genome sequencing, and that was done using sequence annotation method. Basically, this is Frederick Sanger's method. By using Frederick Sanger's method, this was being done. So please know about this also. Got it? As I'm telling you, there are less chances for the same questions to come in. Similar concept will be coming in. Make sure you're pro it at GP and everything out there. Got it? Shall we go ahead? Now, name the evolution of any two plant forms through geological periods from tracheophyte ancestors. By the way, was that the other question clear to you guys? If yes, can you just give me a quick thumbs up? Yes, thank you, Anbugauda and Hassan. Okay, ma'am, please tell me, Lakshmi. So there is no private video, you have to go through YouTube only. Yes, Prithvi. Okay, perfect. Listen, you are being asked about evolution of any two plant forms through geological periods, right? To tracheophyte ancestor. First of all, you need to understand how did plant evolve? So listen to me carefully. The first one were algae, the primitive plants which were there. Then you got bryophytes. They had root-like, leaf-like and stem-like structure, but they were not exactly looking like a plant. Then you had got pteridophytes. You know what are pteridophytes? Pteridophytes are organisms which for the first time they got the vascular tissues, as in the xylem and phloem. Okay. Then from pteridophytes you got your gymnosperms. From gymnosperms, what did you get? You got angiosperms. That is the flowering plants. So please remember the flow. Now we go ahead and see this particular thing. Okay, ma'am, 3 points enough without explanation for this particular thing. You just have to give them, right, the evolution pattern, that's all. Nothing else. So over here, if at all I see, I can just talk about from tracheophyte ancestors, I need two plant forms, right? So tracheophyte ancestors is this. First of all, chlorophyte ancestors means algae. Okay. In between chlorophyte and tracheophyte. Tracheophyte means tracheids. Tracheids are found in what? Pteridophytes. So basically these are the ancestors of pteridophytes. In between pteridophytes as well as algae, what did you have? You have bryophytes. So basically bryophytes goes up over here. You see that? Now from tracheophytes you have to form pteridophytes, right? So tracheophytes, then Rhinia type plants, then xylophyton, then you can write from xylophyton you got what? Spinopsids, or from xylophyton you got ferns. If at all you write tracheophytes first, then Rhinia type plants, then xylophyton, and then ferns, you are basically showing that this is how pteridophytes evolved. Next thing that you can probably show over here is that tracheophytes, Rhinia type plants, xylophyton, and from xylophyton basically you got conifers. Or basically you can say you got ginkgos. Ginkgos, conifers, needles, cycads, they all of them, my dear children, they are gymnosperms. So basically if it goes like ferns and spinopsid, you're talking about pteridophytes. If it goes like conifers and ginkgos, or basically needles and cycads, you're talking about what? Gymnosperms. So it will be easy for you to remember it like this. Okay, it will be easy for you to remember all these things. Now, if you're talking about angiosperms, for angiosperms, the route is pretty much longer. The angiosperms, first tracheophyte, then Rhinia type plants, then xylophyton, then progymnosperms, then seed ferns, then dicots, then monocots are there. Dicots and monocots. This is how it has to go. So basically, you just need to write the easiest one, and the easiest one is this only. This, and then ferns. Or probably this, and then conifers. Okay, please remember ginkgos, conifers, needles and cycads, they are gymnosperms. These two guys, they are what do you call, pteridophytes. Okay, and where were they dominant? In the Mesozoic era. When in the Mesozoic era? In the Jurassic and Cretaceous period. Jurassic and Cretaceous period. You will be asked the order of it also. So please know the order. The CC ones are over here. The GG ones came late. CC ones came first. GG ones came late. Over here, ferns came first. Spinopsids came late. Please know that. Jurassic first, Cretaceous second. This is Mesozoic era. And then over here also, at the end of Mesozoic era, you got the dicots. Dicots and monocots comes under angiosperms, right? So here you got the angiosperms. First one guy to come was dicots. Then in the next phase, that is Cenozoic era, you got the monocots. Okay? Cool? Works? Is there a need to remember the period of an organism? Yes, you need to remember their period. This is the one that is usually mostly asked. So please remember their period. So everyone, I'll ask you a question. Conifers, ginkgos, cycads and needles, in which era were they dominant? In which era were they dominant? Paleozoic, Mesozoic or Cenozoic? Paleozoic, Mesozoic or Cenozoic? When do you think ginkgos, conifers, needles and cycads were dominant? Mesozoic, exactly. It will be Mesozoic. In Mesozoic, which part? It is Jurassic and Cretaceous. Jurassic and Cretaceous. Towards the end of Cretaceous, dicots came in. After that, in Cenozoic era, you got monocots. So please remember all this thing. Very, very important thing. Ma'am, 3 points enough without explanation. Which question are you asking, dear? Which question are you asking? Ma'am, question number 24. Acha, sorry. Question number 24. Just a second. Yeah, actually this is a 2-marker question, so even if you write 2 points properly with explanation, that would be more than enough. So please explain a bit, that is going to be good. Okay? Bye ma'am, Isa paining, oh my god. Adnan King, please take care of yourself. Fine. Children, can I go ahead if this is clear? If it is clear, Janu, you just give me a thumbs up. Yes. If it's clear and we can go ahead with the next question, just give me a thumbs up. Yep, miss. Thank you so much, Hassan. Okay, listen to me. Yes, ma'am, clear. Lakshmi also. Ma'am, me. Yes, tell me, dear. Preetam, Saida. Thank you. Okay, listen to me carefully. Describe briefly the primary treatment of sewage. That means you should know all the treatment plans of sewage. That is what you mean by this, because you can get any question from the sewage. It can be primary treatment, it can be about flocks, it can be about BOD, it can be about secondary treatment, it can be about any tank out there. So please know about this. Class timing, dear, it has started at 12 o'clock. It will end, I think, probably by 6. Maximum is what I am talking about. Okay. So over here, guys, when you're asked about primary treatment, what do? In primary treatment, the simple thing is that you put all the things into a settling tank. What tank? Settling tank. In the settling tank, what is going to happen? Basically, whatever things can float, they will be floating around. Whatever thing has to sediment, which is of high weight, it will be sedimenting, right? And whatever thing is left behind, it will be known as the primary effluent. The primary effluent. So listen, the process of removal of the things which are floating is known as filtration. Is known as filtration. The process of removal of things which are sedimenting down is known as what? Sedimentation. Is known as sedimentation. So you did filtration, you did sedimentation, you removed this. Now what is left behind? Primary effluent. This is sent for the secondary treatment. Until now we were doing the primary treatment. Where was it done? In the settling tank. Okay, where was it done? In the settling tank. Yes, MP Myler, it involves sequential filtration and sedimentation. So over here, primary treatment of sewage is the first physical step in the wastewater treatment where large and small solid particles are removed. Floating debris is removed by filtration. Grit, sand, soil, pebbles is removed by sedimentation. The settled solids form the primary sludge, whereas this particular solid which is there is known as primary sludge. You do not need it, you remove it, okay? The sludge which is there, you remove it. The upper clear liquid is known as effluent. This effluent is sent for the secondary treatment. Okay? Hi, Syed. Okay, guys, I have a question to you guys. I have explained the primary treatment over here. Do you want me to explain the whole sewage treatment right over here, because you will be seeing questions on the basis of it. So do you want me to do it entirely together, or do you want it like whenever the question comes you want to solve it? No, ma'am. So yes, ma'am, no, ma'am has come. Yes, ma'am, no, ma'am. Oh my god, no, 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 no. So no is more and yes is less. Okay, fine. Whenever the question comes, fine. Let's do that then. Let's complete one particular paper then. Fine. Now differentiate between ex situ and in situ conservation. Very simple. Very, very simple. Okay. Ex situ conservation is what? You are going to conserve organism outside their natural habitat. For example, zoo. For example, botanical gardens. That is ex situ conservation. Okay, that is not their natural habitat. You are conserving them over here. What about in situ conservation? You are going to conserve organism in their natural habitat. Okay, so over here, what is the difference that we have over here? In situ conservation, conservation of species in the natural habitat. Ex situ, conservation of species outside the natural habitat. Okay, in situ, what are you going to do? You are going to conserve the entire ecosystem. Why? Because basically, I am conserving a forest and all the organisms who are inhabiting are also getting conserved. So am I not conserving the entire ecosystem? Yes. But over here, I am conserving only the selected organisms out there. Okay, so which is why this is your answer. Now, differentiate between ex situ and example is what? Wildlife sanctuaries and national parks. Wildlife sanctuary and national parks. Here it is zoo, botanical gardens and seed banks. I want your attention over here and I want an answer. Listen to me very carefully. Okay. And that is this. Give me other techniques in ex situ conservation apart from zoo and botanical gardens, because this is pretty much famous. Zoo and botanical gardens are pretty much famous. I want some other technique also which is used in ex situ conservation, the advanced ex situ conservation techniques. Anyone? Ma'am, when is Comet K last date? Neeraj, it is on 16th of March is what I remember. Cryopreservation is what Sayada is saying. Cryopreservation. How does cryopreservation becomes an advanced ex situ conservation technique? Listen, over here, why it is advanced is that it is not you're conserving an organism out there. No, you are conserving their gametes. You're conserving something out there which can be useful to make sure that this organism is not now extinct. You are conserving it outside their natural habitat, yeah, in minus 196 degrees Celsius in liquid nitrogen. So this is one advanced technique. Apart from that, in vitro fertilization. Yes, in vitro fertilization is also something. IVF. Inside the laboratory condition, you are fertilizing with the gametes that is there. Again, for example, some organism were getting extinct. Now you took the gametes and you're fertilizing. Basically, that organism is not now extinct now. So you did IVF is also one thing. Next example, seed culturing. You have a better name for it. There is one more name for it. You call this as seed bank. Seed bank is basically, again, you're conserving the seed outside the natural habitat. You're conserving it such that it does not get extinct. These techniques are super important. They ask you the advanced techniques of in ex situ conservation. Apart from it, one more thing is there. The last one thing, if at all you say that it's done. Ma'am, you're teaching like a best friend. Oh, say hi to your best friend. And Yes, tissue culture, exactly, Mansi. It is known as tissue culture. So what is tissue culture? Basically, you take a part of the plant which is able to divide and you make multiple copies of it. Again, when you're doing it, do you think you're doing it in the natural habitat? No, it is occurring naturally. No. So basically, all of them are coming under ex situ conservation. Do not forget it. Tissue culture, cryopreservation, IVF, seed bank, ex situ conservation. Ex situ conservation. Super, super, super important. Okay, now apart from that, over here, okay, one more thing. Apart from zoological parks and botanical gardens, there is one more thing that you have to tell. Apart from zoological parks and botanical gardens, one more type of thing is there where you preserve organism out there. Any idea? It starts with wildlife. It starts with wildlife. But you do not call it wildlife sanctuaries, because wildlife sanctuary come in situ. There is something else related to wildlife, and children do the mistake over here. Wildlife safari parks, exactly. It is known as wildlife safari parks. So whenever you think you get wildlife safari parks, please understand it's ex situ. Wildlife sanctuary, national parks, everything is in situ conservation. Okay, apart from it, you have one more thing. What was it? Sacred groves. Sacred groves also come under this. Cool? Cool. Yes, perfect. And your biodiversity hotspots also come in. How many biodiversity hotspots are there in our world? How many biodiversity hotspots are there in the world? Any idea? 3, in the world I'm asking. 34. It's not 32, it's 34. Okay, 34. 34. India 3. In India 3, but in world 34. Remember that. Okay, perfect. Let's go ahead. Guys, first of all of you tap your back like this. Say awesome, awesome, because we have completed all the 1 markers and 2 marker question of MQP1. And finally, we enter what? We enter the next thing. That is the 3 marker questions out there. 6, 7, 6, 7. It's not 6, 7, dear. Sorry, it's 34. Okay. 25 was there earlier. Yes, 34, but then 7 were added up. Done. Guys, hi Anjali, hello, hello, hello. Okay. Ravi Chandra, you should not be asking about just passing in biology examination. Listen, I have put in so much efforts. If at all you guys are just thinking I'll just pass in biology, I do not know what to do. Go watch the videos, okay? Okay, so I think there is some double meaning stuff you put in and I didn't realize it because you guys are Gen Zs and I'm different. Okay, fine. Anyways, let's go ahead. Draw a neat labeled diagram of monocot embryo. What do you mean by that? Che, what do you mean by that? What is the question? So basically you have to draw a diagram of monocot embryo. How do you draw that? That's the question. So listen to me very carefully. Monocot embryo. Embryo has an embryonal axis, right? So it looks like this. Embryonal axis looks like this. There will be plumule over here. Plumule is what? Something that is going to give rise to a whole shoot out there. And there will be radicle over here. What is radicle going to do? Giving rise to the whole root out there, right? Since it is a monocot, you need to understand that monocots just have one cotyledon. So there will be only one cotyledon. And what is this cotyledon known as? This cotyledon is known as scutellum. This cotyledon is known as scutellum. Okay, and also remember, monocots just have one cotyledon, which is why they have some endosperm left behind. So usually endosperm is something that will be helping in the formation of cotyledon. In some organisms, since only one cotyledon is being formed, you will have some endosperm still left behind. So that is the endosperm which is left behind. Now since it is a monocot organism, you need to understand that plumule has a covering around it which is known as coleoptile. It is known as coleoptile. Similarly, radicle also has a covering around it. First covering is known as root cap because it has to go inside the soil, and apart from that it has another covering which is known as coleorhiza. Coleorhiza. So here it is R for radicle, there it is P for plumule. Okay, now okay, so over here, if at all I want to make this particular diagram of a monocot embryo, how am I going to do that? How will we do that? So listen to me. It's a very simple thing. Make a whale's face over here. This is the first fin that we have. Whale has a tail. Tail goes like this. And then you have the second fin which is there. What is this fin? Actually, this is the scutellum. That is the first cotyledon that you have. The second cotyledon also was tried to be made, but then it didn't happen.
Okay, it got blasted off. So you call this as epiblast. Okay, this is a rudimentary cotyledon which was not able to form itself, right? Now over here, you have to make something like this. This is going to be a triangle thing. This is what, Baba, okay. And then again, you have to make a triangle thing. So inside this, you have the shoot apex, which is basically known as what? The plumule. And surrounding the plumule, what do you have? You have the coleoptile. So this is going to be the coleoptile, right? Now, you have to join this and go inside and come back and go to the one tail, only one tail, and then come back and join over here. This particular thing which is going to be there is known as a radicle, and as you all know, radicle is covered by something that is known as root cap. Again, this is also going to be covered by something else. What do you call this as? Coleorhiza. Coleorhiza. So please know this particular diagram. Okay, this particular diagram is super important. Okay, just a second. Okay. Ma'am, which video should you see to get good? So, dear, I have, okay. Guys, by the way, I hope this particular picture is clear. If it is clear, just give me a thumbs up. And what was I saying? I was saying something. Which video should be good? Yeah. So, one marker, I have done a video, all 1 marker, all 2, 3 marker, and all 5 marker will be released. I have recorded already. These videos should be more than enough because I have included model question paper questions, I have included question bank questions, HOTS questions, everything. So all 1 marker, 2, 3 marker and 5 marker is important. Okay, you can go check it out on the playlist. In the playlist, there will be 2nd PUC Biology Endgame for separately. You can just check out the videos in there. Dear guys, children, I will be giving you a break once this particular model question paper is done. Can you bear with me until then? Is it okay? And thank you, I will be drinking water. Perfect. Let's go ahead then. Also, this question can be framed in a different way. Instead of asking you monocot embryo, they can ask you grass embryo. They can ask you a grass embryo. So please know that. Dicot embryo diagram. Okay, dicot embryo diagram is super easy. I'll tell you how to draw it. See, see, it goes like this, okay? Control Jasmine. Okay. So you need to make again the plumule and the thing over here, embryonal axis which is there. Here dicots will be having 2 cotyledons, right? 2 cotyledons. So one cotyledon goes like this. You can imagine like that. One cotyledon is this. Another cotyledon which is there, it also goes like this and join it up. This is the 2nd cotyledon. Now over here, these 2 are the cotyledons. Mentioned that they are what? Cotyledons. What is this? You know this is plumule. What is this? This is going to be radicle. Radicle is covered by something always that is known as root cap. Do they have coleoptile and coleorhiza? No. Now the portion below the attachment of cotyledons, it is known as hypocotyl. Hypocotyl, where you have your radicle and stuff, right? The portion above the embryonal axis joining is known as epicotyl. And that is where you have the plumule. And this is your dicot embryo diagram. Okay? Fine. PDF, dear, you will be getting it. Ma'am, your diagram is really better than mine. Thank you. No one says that, but yes. Okay. Perfect. Let's go ahead. Now the next question out there. There is question number 29. What is the menstrual cycle? Mention 1 pituitary and 1 ovarian hormone that plays a major role in the menstrual cycle. Ma'am, PDF fair. Again, if at all you go down, scroll in the description section of this particular video, you will be able to see to download lecture notes. Okay, there will be an option to download lecture notes. I'll be giving you that. Just a second. Chumma, my time and your time will be wasted, right? So let me do that. Ayo. Yes, Shreyas, I can. Okay. Fine. Now, guys, do not disturb me. Okay, do not disturb me. We are friends, but we are studying right now. Okay, fine. So this is like the group lecture where no one studies. So don't make it go like that. So I am the teacher. Please, I am reminding myself that I am the teacher. I do not have to make fun. So over here, please bear with me. Let's do the question. We'll talk about the menstrual cycle and also the hormones which are there up there. Okay, which plays a very important role. So always remember, you have the cycle which goes around like this. This is the first day. and this is going to be the 28th day. Why 28th day? Because usually a menstrual cycle runs up to 28 days, right? Now over here, listen, the first 5 days which are there, it is known as the menstrual phase. Why the menstrual phase? Because over here, your endometrium which was being formed, it is going to shed down. It is going to shed down. Why do you think endometrium sheds down? Because the two hormones which maintains the endometrium, their level is going to be super slow. Everyone who is watching this video, make sure you listen to me super carefully. This is super important. If you get the track of menstrual cycle, it will be super easy for you. Okay? Listen to me carefully. So over here, the two hormones which maintains the endometrium is estrogen and progesterone. And both of their level is less. Who releases it? They are the ovarian hormones. They are the ovarian hormones. Ovaries are going to release it. Now what do you have? You have the next set of things. You have your pituitary hormone. Pituitary gland also releases some hormones, FSH and LH. Follicle stimulating hormone and LH. Listen, these two guys which are there, they are enemies. When one goes up, another goes down. Okay, when one goes up, another goes down. So please listen to me carefully. Estrogen and progesterone, when their level goes down, slowly FSH and LH are going to increase. And during this phase, when they are increasing, we get ahead into the next phase, which is also known as from the 5th to 14th day. Why 14th day? Because this is the middle of the cycle. This is going to be the middle of the cycle. 14th is the middle of 28-day cycle, right? And what happens on the 14th day? Ovulation is going to happen over here. Ovulation happens over here. So basically from the 5th to this particular days out there, we say we are entering into the next phase, which is known as follicular phase. Why follicular phase? Because your primary follicles, exactly, follicular phase. Why it is happening? The primary follicles which are there over here, they are going to mature and become the Graafian follicle. What? The Graafian follicle out there. Okay, and these follicles, you have to make sure you understand this. Here the FSH and LH was high only until the starting point. Now these follicles are going to release something that is known as estrogen. So the follicles are something that is releasing what? Estrogen over here. Since estrogen is being released over here, what is going to happen? The endometrium which was shedding down, it will slowly start regenerating. It will slowly start regenerating. I'm not saying it becomes very thick and things like that. It just becomes regenerating, which is why I'll also call this phase as proliferative phase, as someone was saying. I see Saida was saying proliferative phase, right? So this is also known as what? Proliferative phase. Cool? It is known as proliferative phase. Now listen to me carefully. Estrogen level is also going high when the follicles are developing. The FSH and LH, basically it was high, but now when estrogen was there, it went little bit low. But the thing happens, the something that is totally unexpected happens over here. That is, when the estrogen level goes high very much, at this particular point of time, LH and FSH also shoots up. LH and FSH also shoots up, especially the LH. It was going down, but now it is going high. Especially the LH, it shoots really high, which leads to what? LH surge. When LH surge happens, that's when ovulation is going to happen. That's when ovulation is going to happen. What do you mean by ovulation? Basically, the Graafian follicle is going to rupture and is going to bring out what? The secondary oocyte which is there. Okay, this is known as ovulation. Fine. Now, after ovulation, ma'am, what is that known as? We enter the next phase, which is known as luteal phase. Why a luteal phase? Because the LH which is there, which is causing ovulation, it causes one more very important thing. Whatever thing was left behind. So when I say the secondary oocyte is being released, whatever thing is being left behind the Graafian follicle, they are going to make something. What is that something? They will be making something that is known as corpus luteum. And corpus luteum is basically maintained by the LH itself. The LH is the one that is going to maintain the corpus luteum. That is why it is known as luteal phase. Okay, and MP Myler also said it is known as secretory phase. I also said it is known as secretory phase. Why secretory phase? Listen to me carefully. This corpus luteum which is there, in turn it is going to release progesterone. So until now we didn't study about progesterone. It was going down, then estrogen came into picture. Progesterone was never being studied. So progesterone and also estrogen, Both of them are being released by corpus luteum, especially progesterone, which is why now what will happen, the endometrium which was there, which was just regenerating, it becomes very, very thick. It becomes very, very thick, and not just that, it is going to start secreting some very important things. Why secreting? Because my egg is released, right? The egg is being released, sperm can come any time, and when the sperm and the egg meet, fertilization can happen, zygote can be formed, embryo will be formed. There should be someone to nourish this embryo. And what is that? Endometrium does that. So endometrium is going to secrete some glycogen and stuff like that, which is why you also call this as what? You also call this as secretory phase. Secretory phase. This is super, super, super important. Who does that? Progesterone is responsible for that. Now listen to me carefully. Suppose fertilization didn't take place. Okay, that means there is no point of having an endometrium also. Slowly what is going to happen, usually progesterone which is there, it does one very important thing. It says that I cannot see this guy, make sure this guy is not over here. This is my enemy, I do not want to see him. Basically when it says that, or basically when it gives signal to your hypothalamus or things like that, what is going to happen? The LH level is going to go down. If LH is going down, corpus luteum is not going to be there. If corpus luteum is not there, progesterone, estrogen also goes down, and that is exactly how the same thing repeats up. Got it? This is super important for you. Very, very important. So first, two ovarian hormones are estrogen and progesterone. What is it estrogen responsible for? Estrogen is responsible for, ultimately, for regenerating the endometrium, and also when its amount increases, it is leading for LH surge. LH surge leads to ovulation. That is why we need estrogen. What is the role of progesterone? Progesterone is reached over here in this particular phase where you want to thicken the endometrium and you want the implantation to happen. Okay, that is the role of progesterone over there. It is going to secrete, help in secretion of certain things that will be helping the embryo to form. What is the role of FSH? FSH help in stimulating the follicles such that the follicles become mature. Okay, and this FSH secretion makes the follicles mature, and the follicles is going to release what? Estrogen again. Now what is the role of LH? LH increases its amount, and LH surge leads to what? Ovulation. And LH is important for maintaining corpus luteum as well. You get it? So all this hormones list is very, very important. Okay, please make sure you go through this. Is this clear? Because if I remove this, it won't be given to you back. So if at all you want to take a screenshot, you can take a screenshot. Okay, this is a new page that I've added up. If it's clear, can you just give me a quick thumbs up? Ma'am, please give all 3 mark questions and answer PDF. There, it's there in the WhatsApp channel called along with the video link. Everyone? Yes, ma'am, clear. Thank you so much. Perfect. So I'm just moving it back. So how do you write the answer for this? What is the menstrual cycle? Menstrual cycle is the monthly cyclic change in the ovary and the uterus that occur in the human females for about 28 days, starting from the menstruation to the next menstruation. Okay. Now the next thing that we have is functions of hormones in the menstrual cycle. FSH, you are asked about one pituitary hormone. Pituitary hormone is FSH. So you can write about either of them. FSH basically help in growth and maturation of ovarian follicle. And talking about ovarian hormone, you have estrogen. You can just say it is there for repair and proliferation of the uterine lining, endometrium, and also it leads to LH surge. Works? What is FRS and Mrs. diagram? Dear, I didn't get it. Shreyas, Kannada, dear, if at all I start speaking Kannada, probably you guys will run away from me. Okay, so yes, I am still in the learning phase. Otherwise, I would have definitely done that. Okay. Mr. Perfect, hello dear. Okay, let's go ahead. Write any 3 principles to be followed by people to be free from sexually transmitted infections. STIs, again, each and every part of STIs is important. How does an STI cause, or basically what is there for prevention? What are the symptoms of STI? That is all important. What are the diseases that can be cured, that is not curable? Everything. Okay, so first thing that has to be followed is that avoid multiple sexual partners out there. This is this will be increasing the chances of someone to get STIs. Second thing, correct and consistent use of condom. Why? Condom is the only contraceptive that will prevent pregnancy, and it also prevents STIs, because with the help of condom there is no exchange of genital fluids. And if there is no exchange of fluids, do you think STIs is going to be caused? No. Okay, fine. The next thing that we have is prompt medical checkup and treatment. If at all you have a prompt medical checkup, basically you can stop the symptoms being worsened, because you know what, usually STIs are asymptomatic. Later on people will develop symptoms, and by that time it will be too late. Okay, and last but not least, avoid sharing needles. Now, not all STIs are spread through blood. You need to understand that. Only few STIs, for example, I talk about HIV, AIDS and hepatitis B. hepatitis B. Those are the ones which are spread through blood. Okay, so in that case, yes, avoiding sharing needles and things like that work. Otherwise, these are the 3 points that you need to write. Make sure you write the 3 points clearly. The diseases are transmitted by sexual contact between two organisms. Exactly. Now, before I go ahead with this question, I want to ask a question. What are the non-curable STIs out there? What are the names of the non-curable STIs which cannot be cured? Gaurav, hi dear. What are the non-curable STIs? Non-curable STIs. HIV AIDS. HIV AIDS. HIV is a virus, okay? HIV AIDS is the answer, okay. Ma'am, you would have been a doctor. Okay. Hepatitis B. Okay. HIV AIDS, hepatitis B, correct. One more disease is there. Come on, guys. Try Madi. HIV AIDS, hepatitis B, one more thing. Who have been through my lectures very carefully before might know the answer for this, because there is a trick that I always use for remembering it. Gonorrhea. No. You give me all the options, genital wart, genital herpes, everything you give me, guys. Oh, what is this? Machas. Listen to me, machas. So over here, HHH, HIV AIDS, hepatitis B, and herpes, or you also call this as genital herpes. These are the 3 things that you need to remember. HHH, MP mylar, exactly. Okay. Make sure you remember it, it's important. And make sure you know the symptoms also. Can anyone say what are the symptoms of STIs? So what are symptoms of STIs? Come on. Few symptoms of STIs. Why am I doing it? Because I want you to revise it. Very important. Itching. Yeah, itching is there. There will be itching. Apart from that, pain. Okay, pain. Itching will be there, pain will be there. If at all you talk about worst symptoms out there, there is pelvic inflammatory disease. In the pelvic, there will be inflammation out there. Fluid discharge is there, exactly, MB mylar. Okay, fluid discharge, itching, pain is there. Most of the ladies who would be having it might be asymptomatic later. They might have developing symptoms, such as I told you, pelvic inflammatory disease. Apart from that, ectopic pregnancies. Ectopic, this is not what do you call it, symptoms. These are basically the problems that are associated with STIs. It can lead to ectopic pregnancy, as in pregnancy in a place apart from uterus. It can be in the fallopian tube and things like that. Apart from that, what is there? Infertility issues can be caused. Stillbirth can happen. That is, baby will be born, but it will be still, as in it will be dead. Okay, so please know that. Usually it is itching, fluid discharge, as well as pain in the pelvic region. Yeah? Cool. So, yes, let's go ahead. Next question. Draw a label diagram for Stanley Miller's experiment in relation to chemical origin. Perfect. This question is very, and infertility. Yes, Yasmin. Perfect. So this picture and this particular question is one of the favorite because it is very easy to explain. Listen to it carefully. Stanley Miller was trying to prove their friends' theory. Friends were Oparin and Haldane. Okay. Oparin and Haldane, my dear children, is very important. What did they do is that they said that Earth basically had chemical evolution. What do you mean by chemical evolution? They said that there were some inorganic things in the primitive Earth. These inorganic things which were the ones which made the organic things, and these organic things were actually made the first cells out there. The first cells out there. This is how things went. This is known as chemical evolution. Now there was someone who there needed to be someone who could prove this, and that was Stanley Miller. So Stanley Miller, and also one more thing that Oparin and Haldane said, in the primitive Earth, it was very hot. It was very hot. Okay, the temperature was very high, and apart from that, it was reducing atmosphere. What do you mean by reducing atmosphere? That means there is no oxygen. Oxygen was not there. So this thing also had to be maintained. So then Stanley Miller was like, let's check it out with the help of my setup. So this setup was being made where what is there? One more thing is there. Shall I say it now only? Yeah, I'll say that now. Inorganic things. By inorganic things, what do you mean? The inorganic things are methane, that is CH4, not CH3, NH3, that is ammonia, and also hydrogen. Apart from that, there is water vapor also. Water vapor. So apart from hydrogen, there is water vapor also in there. So 4 things you have to remember. 4 things. Okay, now let's do the setup. So this setup was being made where boiling water was being kept. Why boiling water? My dear children, with the help of boiling water, over here, the water vapor came in. If at all you see this whole setup, it is very much closed. That means there is no oxygen in this particular setup. It is reducing environment, right? And over here, you have 2 electrodes. Why do we have 2 electrodes? To maintain the temperature. It was 800 degrees Celsius over here. How much degrees Celsius? 800 degrees Celsius. Now, these gases were also there. As I said, CH4, NH3, H2O, that is in the form of water vapor, and also H2. And later, he saw something, that is, in the water that was accumulating over here, he saw the organic matter. Do you know what was the organic matter that he found? Anyone? Stanley Miller found what organic matter in this particular setup? Can we get these papers? These are model question papers. Yes, you can get the PDF of it. Please join the WhatsApp channel. Yeah? Guys, what do you think was the organic matter that he got from this experiment? Amino acid. Exactly, Paru. It was what? Amino acid. This is super important. So this amino acids, my dear children, they will come together to form proteins. These proteins can come together to give rise to a whole new cell along with other things, right? So according to Stanley Miller, he did this experiment and he got only amino acid. There were other people who did the experiment and they got nucleic acid, they got sugars and things like that. So you should know, in Stanley Miller experiment, only amino acid, nothing else. Okay, please know that. No sugar, nothing apart from amino acid. Others did the experiment and they got multiple other things. Got it? Is this question clear? If it is clear, can you just give me a quick thumbs up? Everyone, we'll go on to the next question. Quick thumbs up. Love you, ma'am. Love you too, dear. Okay. Thank you. Perfect. Prithvi, thank you. Prachi. Okay. Next thing that we have is name the causative organism of the following disease. That means you can get a question based on this also. So how do we explain that? Filariasis, ringworm and malaria. You have to tell the causative organism of all this thing that is there. Any idea? Any idea what do you think is the answer for this? So, listen, filariasis is caused by what? Wuchereria bancrofti. It is caused by a filarial worm which is known as Wuchereria bancrofti. Apart from Wuchereria bancrofti, there is also V. malai. Apart from it, we have Wuchereria malai as well. The next thing that we have is ringworm disease. Ringworm disease, it sounds like it is caused by a worm, but it is not caused by a worm. It is actually a fungal disease, which basically on our skin looks like a worm is there in the round format, but it is not a worm, it's fungal disease. It is caused by 2 people who are fighting, trichophyton, epidermophyton. Trichophyton, epidermophyton, my dear children, they are all fighting. Why are they fighting, ma'am? They are fighting for a small spore, which you call as microsporum. So basically, you can remember like that, trichophyton, epidermophyton and microsporum are the fungus that basically, fungal species that causes ringworm disease. Now, we are left with only one thing, that is malaria. Malaria is caused by 3 guys out there, okay? Yes, oh my God, all of you are writing the answers. Perfect. Perfect. Make sure you know the spellings. I see the spellings are also wrong over here. Don't get your spellings wrong. Okay, don't get your spellings wrong. Now, over here, what do we have? Malaria. Malaria is caused by Plasmodium vivax, Plasmodium falciparum, Plasmodium malariae. Out of these 3 causative agents of malaria, can you tell me which one causes the malignant form of malaria, the dangerous form of malaria? Anyone? Yeah. Perfect, Jia, Prachi, and Plasmodium ovale is also there, not there in your NCRT. So, yeah. P. falciparum is the one that causes the malignant form of malaria. Always remember, this is the one that causes malignant form of malaria. Perfect. Hello, Vasik. Okay. So, my dear children are super awesome. Is that what I get from this? Perfect. Let's go ahead then. Next question that we have is from the chapter Microbes and Human Welfare. These are, by the way, children who are joining up there. I hope you guys know this is model question paper. Hello, Shreyas. I'm seeing you. Okay. Next question is, mention the uses of the following bioactive agents: streptokinase, cyclosporin and statin. One by one, everyone tell me. I'm not going to show you in that way. Streptokinase, what is the use of that? First one, streptokinase. What is the use of streptokinase? Gia, what is your fave? Okay. Okay, Shakira says the answer for this is clot buster. Prachi also says clot buster. Okay, clot buster. Oh my god. Mansi, Harshik, everyone is saying clot buster. Yes, streptokinase is basically a clot buster. So basically, if someone is having a myocardial infarction which is leading to heart attack, there might be clots in their body and you have to remove the clot. And that is the role of streptokinase. Can someone tell me where do you get this organism? Where do you get this enzyme from? Where do you get this enzyme from? Streptokinase you get from which organism? Prithvi, Prachi, everyone, correct. Streptococcus, exactly, that is a bacteria. You need to remember that it is Streptococcus from where you get the streptokinase. Now the next thing that we have is, so this is an enzyme, remember, in your NCERT, this is an enzyme. And then you have been asking about the bioactive molecules. The first bioactive molecule is cyclosporin A. Cyclosporin A. First tell me the function. What is the function of cyclosporin A? What is the function of cyclosporin A? And by the way, all of you, Shakira, Prithvi and Meenakshi, hello dear, Saida, Harshika, Jia, Sneha, perfect, Yasmin, Hassan. Immunosuppressive agent. Yes, it is an immunosuppressive agent. Why do you need an immunosuppressive agent? When do we need an immunosuppressive agent? Listen to me carefully. Suppose I am having, my kidney is gone. I am taking a kidney from Hassan. Okay, my body has high chances for rejecting it. Why? Because it is foreign thing. Which is why, basically, even after doing a whole surgery, I will not be able to survive. Okay, the thing is that in that particular case, I have to make my immune system suppress a bit. My immune system is super active. So I need immunosuppressants, okay, during organ transplantation. Exactly. Now tell me the organism from where you get this cyclosporin A. The organism from where you got, Shreyas gave me the answer for the next one also. Trichoderma polysporum, exactly, Prithvi. Trichoderma polysporum. Trichophyton, your Hassan, previous time also, same mistake, I still remember. It's not trichophyton. Trichophyton is basically in fighting thing. That is ringworm disease. Over here they are fighting. Okay. This trichoderma. Trichoderma polysporum. Trichoderma spelling, please check Madi, everyone who is giving me the answers. Trichoderma polysporum. Okay. Ayyo, my bad. It's okay, just remember. So you can remember, if at all you're finding it difficult, Hassan, to remember this, listen to me carefully. Trichophyton, epidermophyton are fighting. Since they are fighting, this is a battleground. And basically that is why we have skin lesions out there, because they are fighting over here. Okay? Cool. Now we have one more thing, that is, statin. So Shreyas already gave me the answer for this, but I still want everyone to try. Statin, what is the use of statin, Baba? Everyone, please try. Shreyas, you are from which district, ma'am? So I was born and brought up in Indore, Madhya Pradesh, but my roots are from Kerala. Okay, now my parents are in Kerala, so yeah. But as of now, we are in Bangalore. So there's like multi-city thing out there. Yes. Blood cholesterol lowering agent. Exactly. Blood cholesterol lowering agent. You can remember like this, statin stabilizes the blood cholesterol. Where do you get it from? I have seen so many people answering that also. You get it from Monascus purpureus. Monascus purpureus. Monascus purpureus gives us what? Statin. So S for statin, Monascus purpureus ends with S. So you can remember like that. And one very important thing, guys, out of them, which one is a bacteria and which ones are the fungus? Which one is a bacteria and which one is the fungus? Very important thing. NJ, hello Mallu. Mallu is also here, or just Malu, I don't know. Zainab, hi dear. Listen, these two guys which are there, Trichoderma polysporum as well as Monascus purpureus, they are fungi, okay? And this guy which is there, this is the only bacteria that we have. These two guys are fungus, please remember. Trichoderma and Monascus purpureus, both of them are fungi, and basically one is going to be bacteria. Know this as well. Cool? Perfect. So let's go ahead. You have given you the answer over here. We have discussed this in a detailed manner. I hope you do not forget it. Okay? Can you visit Belgaum? Surely. I have one colleague over here. He invited me at his home once. So yes, surely once, if at all it happens. Yes. Now describe the steps of decomposition that operate simultaneously on the detritus. So basically you are being asked about the steps of decomposition. Guys, can you try the steps of decomposition without seeing? The steps, first step of decomposition, one by one if at all we go. FLCHM, FLCHM, okay. FLCHM stands for what? Let's see. So let's go and see. F, L, C, H and M. Listen, the first thing is fragmentation, right? What exactly happens in fragmentation? Everyone is correct. Correct. Awesome. What exactly happens in fragmentation? Basically, the bigger things have to be fragmented down into small things. Listen, this is a physical breakdown. Over here, you are not using decomposers. Rather, someone else is coming into the picture. What is that something else? It is basically detritivores. Detritivores are ones that feeds on detritus, that is dead and decaying matter. So here the detritivores which is there, the best example for that is earthworm. They are not breaking down organic things into inorganic things. Listen, the ultimate aim of decomposition is to do what? To convert the organic material to inorganic things out there. We are made up of multiple inorganic things which is now organic in our body. We are breaking it back to inorganic such that new things can be formed, right? Over here in this particular process, it's just that the organism eats up the big thing and poops out the small thing. It is just fragmenting. Now in this particular process, what is going to happen? Some things will be there. Some things will also be formed over here, which is inorganic in nature. Okay, inorganic in nature, and it is insoluble. Sorry, it is soluble as well. It is going to be soluble as well. So before doing the entire process, some things might be inorganic. Okay, these inorganic soluble things can be going into the soil and staying up there. Okay, it will be staying up there. This process is known as leaching. So when at the end product of fragmentation, we get some inorganic soluble things, they go into the soil and they're just there. This is known as leaching. Okay, this is known as leaching. Now what? So leaching is water soluble inorganic nutrients go down to the soil and precipitate as unavailable salt. Exactly, perfect. Now listen to me. This is the time where actually the organic things are going to be broken down to inorganic things. So I can basically make the graph start from here. The organic thing actually starts breaking down from here at this particular point. This is known as catabolism. The breakdown of organic thing into inorganic things with the help of decomposers. What are the usual decomposers? Fungi and bacteria, Facebook. And in pond ecosystem, you have flagellates as well. In ponds, you have flagellates as well. Anyways, the decomposition process has started off. Listen to me. It's not a one-step process. It is not a one-step process. Basically, what happens over here is that, suppose here you get inorganic things. In between that, you form something, and that is known as humus. What is humus? Humus is basically dark amorphous solid substance out there, and this dark amorphous solid substance has inorganic things. It is slowly breaking down into inorganic things. It has not completely, it is slowly in the breaking down process. You get it? So humus which is there, it is dark amorphous thing. It is not acting on anything. It is something that is formed in between, that is known as humus. And this process of formation of humus is known as humification. Listen, I'm telling you again, it does not act on any detritus. It is a process that is happening by the end of which you get humus. Now this humus slowly gives little amount of inorganic thing. And this process, when we say that finally the inorganic things are being released, this is known as mineralization. Mineralization, that means the final breakdown of the things into inorganic things. Okay, the release of inorganic nutrients due to degradation of humus. Exactly. So people consider this to be as multiple steps, but please know, they're all related. It is actually coming under catabolism only. But when you're asked about the steps of decomposition, you have to start from here and then go until there, because obviously these things have to happen for the next things to happen. Okay, and humidification is the process where there is no role of acting on detritus. Okay, please remember that. Is it clear? I hope it's clear. Now, one more very important thing that I want to ask you guys is that, what are the favorable conditions in which, what do you think that decomposition can easily happen? You need a moist atmosphere. You need a moist atmosphere. Now what do you need? You need aerobic condition. Why aerobic condition? This is very important because only aerobic bacteria are the ones that are going to do decomposition. So this is very, very important. Please remember it. Apart from that, if the organic matter that we are breaking down, if at all it is rich in NC, okay, it is rich in NC, that is nitrogen and carbon, it is going to be all the more good. It is going to be all the more good. So these things are something that you need very, very much. Okay? Cool? And over here, what is something that you do not need? If at all the temperature is too low, that is too cold, it cannot happen. Second thing, if at all anaerobic, that means the organism that can sustain in no oxygen condition, they will be coming and doing decomposition, that will also not happen. Similarly, if at all the organic content that we are trying to break down is rich in lignin and chitin, it will be a very slow process. If lignin and chitin is rich, then it will be a very slow process. So this is something that is not favorable, this is something that is favorable. Always remember that. Why no one is teaching Hindi is what I saw a comment by Deepti. We will be teaching Hindi, okay? We will be teaching Hindi towards the examination, and yeah. So just before your examination, you will be getting a Hindi teacher out there for sure. Cool? So we had tried, I know that you needed it earlier, but we had tried it earlier. We didn't get someone who could give time, but we will definitely do that. Okay? Yes. So guys, is this particular question clear to you? Will you be able to answer everything that it comes in relation to it? If yes, just give me a quick thumbs up. Okay, perfect. Thank you guys. So please write all this thing, and yeah. So finally, we have got into the world of 5 markers. Everyone excited? Let's go. Let's go and see all the 5 marker questions out there. Ma'am, full bio syllabus one shot. So full bio syllabus one shot. You know, yesterday there was a recorded video that was premiered live. You can watch that. It is of 10 hours 30 minutes. That video will be definitely helping you well. Okay, let's go. Yes. So draw a label diagrammatic sectional view of the human male reproductive system and label the given parts: testis, seminal vesicle, glans, penis, epididymis, and urethra. This is a diagram that you have to make. Do you want me to make this diagram over here? It's not 11 hours. Actually, at the end, someone did not stop streaming, which is why the 1 hour is extra. It's 10 hours something, 10 hour 20 minutes or something like that. So let me see. Yes, no is saying. So the maximum number of votes, whichever it goes, I'll consider that. Skip, ma'am. No, ma'am, this is easy. Okay. So please, guys, please go ahead. I'll be making a separate video on diagrams also, but please make sure you go through it. Okay? Okay, perfect. Now, next thing. Represent schematically the result of 2 gene inheritance by taking seed shape and seed color in the pea plant. Basically, you're talking about... Gee, I wanted the diagram, huh? Gee, I can show you the diagram towards the end. If at all you wait till the end. Okay? I'm so sorry. I have to go by the maximum votes, yeah? Yes. Yes. So seed shape and seed color, that means you're talking about dihybrid cross. When you say 2 gene inheritance, you're talking about dihybrid cross. This is super simple, and listen to me carefully if at all you not want to lose any of your marks. Let's start with the parent plant. So this is the parent plant. We have round and yellow. And then we have wrinkled and green. How do you write it? These are pure homozygous plants, right? So you write it like this: capital R, capital R, capital Y, capital Y. Here also small r, small y, small y. Done. You make a cross for this. You make a cross for this, right? Now over here, always remember how are the gametes going to be formed. So it is like this: capital R on one chromosome, another capital R on another chromosome, capital Y on this chromosome, capital Y on this chromosome. From this pair, one guy can come in. From this pair, one guy can come in. So it will be obviously capital R only because you do not have any option. Similarly, from here also it will be only capital Y. So there will be multiple gametes being formed, but all of them are going to be capital R, capital Y only. Similarly, from here also it will be small r, small y only. When you cross them, what do you get? You get capital R, small r. You get capital Y, small y. That means what? It will be round and it will be yellow. It will be round and yellow. But then Mendel was like, it is round and yellow like this or like this, heterozygous or homozygous. For that he had to do the cross, right? So let's do that particular cross. That is the F2 generation. Okay, that is the F2 generation. So listen to me carefully. Guys, just give me 5 seconds. I'll complete it. So over, not 5 seconds, probably 1 minute. So it is like this now. It is like this, right? So if at all we make the chromosome, it is capital R, small r, capital Y, small y. Now here, this capital R can go with this Y, because from one chromosome pair, only one guy can go in, right? So here from this capital R, from here capital Y. Similarly, from here again capital R, from here small y. From here small r, from here capital Y. From here small r, from here small r, here small y. 4 type of gametes are possible. You make the 4 type of gametes over here. This particular plant also produces 4 type of gametes. You put it over there, then you cross it. What do you get when you cross it? If at all you cross each of them, capital R, capital R, capital Y, capital Y. Basically, you are going to get 16 combinations. How many combinations? 16 combinations. Okay, 16 combinations are there. And what is the phenotype over here? Phenotype is going to be 9:3:3:1. 9:3:3:1. This is a genotypic ratio. Phenotypic ratio. 9 of them are going to be round and yellow, the dominant parental character. 1 of them is going to be the recessive parent character. What was it? Wrinkled and green. Wrinkled and green. What about the 3 and 3? Let's see. So it will be the new one. Round was with yellow until now. It will be with green now. Wrinkled was with green until now. It will be with yellow now. It will be with yellow now. So basically, can I say 3 plus 3, 6 of the 16, okay, out of 16 out there, 6 of them are going to be non-parental characters. That means new character out there which I didn't see. Yes, I can say that. And why is it happening? Because of independent assortment. Law of independent assortment, which says that from a pair of alleles, only one pair comes in. It has no dependency with the other pair of character. So this is the character for shape, this is the character for color. Both of the genes are going to assort independently. That is why you get multiple type of gametes. Otherwise, capital R will only go with capital Y only. No, that is not happening over here. Always remember it. 1:2:2. Prithvi, check if... Guys. One very important question that I want to ask. You guys all told me that 9:3:3:1, everything is there. How many phenotypes are possible? How many phenotypes are possible in this type of cross? How many phenotypes are possible in this type of cross? How many phenotypes are possible in this type of cross? 4 of them. Is it 4 phenotypes? Someone says 16. 4, no baba. 4. This is round 1, round green, wrinkled yellow, wrinkled green. Right? 4 phenotypes are possible. Okay, how many genotypes are there? How many genotypes are there? Genotypes, genetic makeup. Come on, 16. Gia, remember, previous time, the same question we got stuck. Think. 9. Okay. Sneha, Aradhya and Prithvi is saying 9. Okay, I have to lock 9. Listen to me. There are 16 genotypes over here, agreed. But some of them are going to be repeated multiple times. That is how you get 16. Unique number, if at all you're asking, the genotypes are going to be only 9 of them. Only 9. And you can remember it like this. The first over here is 9. You can remember it like that if at all you want. If at all you want, you can remember like that. Okay, so please do not forget it. This is super important. One more question, last question that I will ask you depending on this. This everyone should be able to do it. Okay, the question goes like this. If at all 64 progenies were there, out of them, how many of them are going to show the non-parental character? If total 64 progenies are there, I have solved this question before also multiple times, but I just want you to ask. I want you to understand. 24. Hassan Sayed's 24. If 64 progenies are there, out of that, how many of them will be non-parental? 32. 24. GIS. We studied the previous time, right? Yes, you remember it. How 24? If at all you are wondering it like that, please remember. Out of 16, 9 plus 3 plus 3 is 1 is equals to what? 16. Out of 16, 6 of them are non-parental. 3 plus 3 is 16. 3 plus 3 is 6, right? 6 by 16 out there. Now, if you're talking about 64 progenies, basically you do the calculation like this. What do you get? You get 24. Out of 64, 24 of them are going to show non-parental. Rest of them are going to show parental character. This is a formula that you can use for that. Works? Okay. How many of them are going to show parental one? If at all you're asked like that. How many of them shows parental character? Anyone? Hello, Tanuj. Tanu. How many of them shows parental? Out of 64, if at all you are asked, how many of them shows parental? Any idea? So listen, 9 plus 1 is equals to what? 10. Now 10 out of 16 into 64. What do you do over here? You do the calculation. What do you get? You get 40. 24 were non-parental. 64 minus 24 is equals to 40. Your answer is 40 now. Think. I asked about 64. If generally you are asking, then it is 10. Yes. Ma'am, classes will be there. Yes, dear. So, yes, class will be there before your biology examination. Okay? Perfect. Got it? So, I hope this is clear. Everyone who feels that, okay, this is clear, I do not have any doubt, just give me a thumbs up. Deepak Hebali, there is no point of just passing. You are going to get good rank. Go study with the videos that is given. Go to a playlist of biology and go start watching those playlists and just sit with those playlists. Imagine that I am your friend and we are both studying together. Okay? Ma'am, cannot download your notes PDF. Is that the problem happening with every one of you? I don't think so. It should be happening because I put it. Can you check, dear, once? Okay. Okay, guys, we'll go ahead. Okay, you realize it that you have not started, then it's high time to start. Yes, next question that we have is DNA is genetic material in both prokaryotes and eukaryotes, yet the packaging process in their cells is not the same. Explain. So DNA is the genetic material in both prokaryotes and eukaryotes, but the packaging is not the same. Why it is not the same? How? Why it is not the same? Okay, why it is not the same? Listen to me carefully. Prokaryotes does not have a nucleus. Do they have nucleus? They do not have nucleus. They do not have membrane-bound organelles, nothing like that. Eukaryotes, do they have nucleus? Yes, Baba, we have a nucleus. So in prokaryotes, there is no nucleus, which is why the genetic material can be, they should not be just fit in. No, there's no problem of fitting things inside something, right? It is very easy for it to be there. Second thing, the genomic size of prokaryotes is very less. For example, Escherichia coli, the genomic size of E. coli, which is there, it is just 4.6 into 10 to the power 6 base pairs. In our case, we are eukaryotes. We have 6.6 into 10 to the power 9 base pairs out there. So genomic size is less. They do not have nucleus to fit in things, which is why it will be very easy. Exactly, Tanu, Ganesh and all those things. Prithvi also. Yes. So how do you write the answers over here? DNA is genetic material in both prokaryotes and eukaryotes, but its packaging differs due to differences in genomic size and also the cell organization. In prokaryotes, DNA is naked, circular, it is circular and present in what? Nucleoid region, because there is no nucleus, right? So do you need histone proteins? You usually need histone proteins to wrap around the DNA such that it becomes compact. So in the case of prokaryotes, always remember there is no histone proteins. There is absolutely no histone proteins because of the small genome and the absence of nucleus. Because of the small genome and the absence of nucleus, right? In eukaryotes, DNA is associated with histones. In eukaryotes, there is a whole packaging difference out there. We have what? We have histone proteins. How many of them? 8 of them are there. Histone octamer. And onto this 8 histone protein, your DNA is wrapped around. How many times? Twice. Twice your DNA is wrapped. How many base pairs are being covered? 200 base pairs are being covered. Over here, you have an additional histone protein, which is known as H1 histone protein. Together, you call this structure as what? Nucleosomes. So basically, we have nucleosomes since we want to fit in the entire DNA into the small nucleus out there. And our genomic content is also very, very big. So please mention that. Ma'am, but it has nucleoid, right? Nucleoid is just a region, dear. It is not a defined thing that has a membrane around it. Okay? Ma'am, tell 5 marker questions.